Researchers looked at different ways of teaching, and what they found is the best way to teach science is to let the students be the scientists. In other words, to put them in charge of asking questions, just like scientists do in research.
One of the groups was so excited about their research that they themselves took their research paper and got it published in a student research journal.
They get so excited, and I really think that's what it's all about, is being able to own their project.
Hello. Once again, we're so happy that you've been able to join us. Welcome back to day 2 of our workshop. And I'm Pam Marshall, a professor at Arizona State University. And it's my pleasure, along with my colleagues, Jennifer Broach, Jennifer Foltz-Sweat, Jennifer Hackney-Price, Kim Kobachek, and Ken Sweat, of welcoming you to day 2 of this workshop, where we're presenting our findings and work on our interdisciplinary course-based undergraduate research experience, something we call an iCURE. We hope that the workshop helps you get started thinking about how to design and implement a cure. So partial support for this work was provided by the National Science Foundation's Improving Undergraduate STEM Education, the IUSE program, under award number 1606903. Any opinions, findings, and conclusions or recommendations expressed in this material are those of the authors and do not necessarily reflect the views of the National Science Foundation. We are excited for day 2 of this workshop. And to remind you, the workshop will be a mixture of pre-recorded videos and live sessions. And the typical flow of the workshop will be pre-recorded videos and then a live Q&A. Please put your questions in the YouTube chat and we will do our best to answer them during the Q&A portion. Thank you again for attending. And now Jennifer Broach is going to talk a little bit about a few housekeeping items.
Thanks, Pam. I wanted to kind of touch base. I really appreciate your feedback from the first day of the workshop. And I am so glad that you guys all enjoyed the workshop, and in particular, the keynote speaker. That was a highlight for most of you. But I wanted to make a few announcements, especially about the keynote and all of the presentations, that they're all going to be posted and captioned. So if you didn't catch everything that Dr. Brunell said, then you have the opportunity to go back and actually see the captions, because I know a couple of the comments was that she was a little bit fast, and because she was trying to give us as much information in a little time as possible. So we appreciate that feedback, but just know that we're on it, and we were already going to caption the videos. And then I will be sharing in the links to the student work, in particular the paper that Dr. Marshall talks about in the intro video with the microbiome paper. We have another paper for the soil remediation, a student paper, and links to student posters, so you can start to see the student work, both the poster work and the actual publication work. So I will provide links to all of those. And we were going to provide a few more little intros so you kind of know what's coming up as the videos progress. So thank you for your feedback. And we have one more survey at the end of today to send out so we can get a little bit more information for the National Science Foundation. Thank you, guys.
Thanks, Jen. So I am so pleased to let you know that the next video that is coming up is the pre-experimental planning and hypothesis formation video by Dr. Jennifer Broach, and then we will have a question and answer session. Thank you.
All right, hi, I'm Jennifer Broach, and I'm a statistician. So I'm going to talk to you today about pre-experimental planning and hypothesis formation. So basically, we want to know and we want to have the students ask the right questions. So why design the experiment in the first place? We want to make sure student researchers design an experiment that allows them to make scientifically valid and objective conclusions. So we really want them to ask the right questions. We want them to collect the right data, and we want them to analyze their data correctly, all in the goal of making scientifically valid and objective conclusions. So all of these activities that are surrounding the statistical aspects of your iCURE are supported from planning to conclusions. So the supplementary materials are provided in all coordinating student activities for your entire iCURE. So we're going to start with the testable hypothesis and factor identification, move on to experimental design, then onto their actual experiment where they're going to collect the data, and then ultimately to their analysis and their conclusions. So we want the students to ask the right questions. So when your students come to your classroom, they're full of ideas. And by now you've done a literature review and the students are starting to generate some ideas that they may want to do for their research project. But students have ideas. They're not necessarily perfect questions yet. So what we want to ask them what they want to learn from their research. We want them to know what question they're trying to answer. So our goal as the leader, as the instructor of the iCUREs, is to get them from idea to a well-defined, testable research question. And so using an analogy from business, we want to make their research questions smart. So the analogous is the SMART goals in business. So we want them to be specific, measurable, actionable or attainable, realistic, and time-bound. Typically, these iCUREs are one semester. We don't want students to come up with an idea that is a multi-year project. We need to make sure that they adhere to some of these SMART objectives. So moving that idea and formulating it into the right question. So a good research question exhibits a couple of these qualities. We want them to ask a question that is relevant and interesting. We also want them to anticipate variability. Their experiment is going to be very, very boring and very uninformative if their answers are all the same measurement on the same things over and over again. You want them to ask a question that anticipates the natural variation. We want them to anticipate that variation in what they're doing. So the other thing is you want them to ask a question that can be generalized by some central tendency, by the number of yeses or the mean. You want them to be able to analyze that amongst groups of individuals. We also want to make sure that they need data. They need to collect data to answer these questions. They can't already know the answer. They have to want to know the answer. They're going to have to investigate this answer. So they need to collect data to do so. And this question doesn't already have an answer. It doesn't actually have a precise, exact answer that is already out there. So again, we want to anticipate variability and we need to collect data. So in this pre-planning phase of this research idea into research question, they're going to go through a whole circle of activities that kind of bring it from an idea to an actual research question, because we want them to start thinking critically and statistically about their research. And we want them to understand the importance of planning and that the planning that they're doing now is again going to help them make those valid and objective conclusions at the end of the day. So we're going to start with the statement and the recognition of the problem. That's generally the idea. But you'll notice that this is a circle. These things happen in tandem. So we're also going to want them to identify the factors of interest, what their experimental factor is, and what they're going to manipulate in their experiment. And then we're also going to want them to select the proper response variable that will allow them to objectively measure the differences in their two groups. So variable selection is of utmost importance when they're setting up their experiment. We want to make sure that they're thinking about how to objectively measure and provide useful information about their research question. So we want to ask them, what are you testing? That is, we're going to add some formality to it. What is your dependent variable? We want to try to constrain our students to using one response variable. We don't want them testing 5 different questions or 10 different questions. We want them to be very focused and specific. Again, back to those SMART ideas. We also want them to note in these pre-planning activities is how they're measured. What is that measurement going to actually look like? Are you measuring yes, no? Is it present? Is it not? Are you measuring it on a scale from 1 to 5, 1 to 10, 1 to a million? You want them to understand what types of numbers or categories or what types of information, what types of data they're going to get from the very beginning, because this is all going to dictate and lead into their analysis. So the second part that we want them to identify is their experimental factor of interest. So this design factor is the factor that they've selected to study. It's that additive or no additive. It's that new drug versus the old drug. It is something still there after the animal was buried. So this is the design factor that the student will manipulate. And these are their independent variables. All terms that your students and yourself, of course, are likely familiar with, but we want to reintroduce them in a very specific way to be able to get them to get to those conclusions at the very end, those objective and valid conclusions. So we also want them to think about what other factors can impact their dependent variable. What other things are at play here? Because sometimes there are factors that you don't really care about, but they are going to impact what happens to your dependent variable. So those are typically called nuisance factors. And we don't have to worry about them too much, but we do need the students to measure them because it's going to impact how we're going to analyze them at the very end of the experiment. So it's important for the student to be able to identify all factors of interest, not just the ones that they're particularly interested in the experiment, but all other factors that would impact that variable. So again, back to the circular motion of this, going from idea to specific research question, I wanted to point out that about 80% of the success in conducting a designed experiment results directly from how well you do the pre-experimental planning. And we want to emphasize this to our students, and we want to make sure that they go through those activities to identify the variables, because these will be used again in every single next step in order to lead them to the proper conclusion. Thank you very much for listening to the pre-experimental planning process. The next process would be the actual experimental design.
Thanks, Jennifer. That was really very good for the students. I can't believe that I never learned anything like that in graduate school. You just kind of hop in and do an experiment and then worry later about how you might analyze it. I'm glad that we are beginning to understand that that is probably not the best way to do science. So now is the Q&A time. So please drop your questions in the chat.
And it's really funny, Pam. I want to avoid the situation that I'm often in as a statistician, where, clients are, I mean, it's not just scientists. So just so you know, it's not just scientists, but they just come to you and they're like, I have data. And they're like, as in, and the very first thing I ask them is, what question are you trying to ask with this data? And oftentimes those two things don't go together, because they don't collect the data that can actually answer the question that they wanted at the beginning. So that's what we're trying to make sure that the students can do, is make sure that they match, that they actually start thinking about the proper data collection for the question that they want to ask, because oftentimes that doesn't get thought about until the end. And then you're like, well, you can't actually answer that. Like, I don't know, like, that's a good question. Let's go out and plan an experiment to actually answer that question, because the data that you have right now doesn't actually answer that question. So I do that. I'm just trying to avoid that. So I think we have a question. How much do you guide students through this process, such as critiques of missing nuisance variables? Do you let them proceed with poorly constrained questions as a learning process or step in? That's a really good question. And I will probably defer to my colleagues that actually go take the students through this, but it's funny because the activity actually walks them through it, and it actually asks them to put the question at the very beginning and then go through the steps of identifying the variables. And then it says, wait, does anything else affect the measurement of what you're doing? Like, could it be the temperature? Could it be some other like external factor? And then it asks them to rewrite their research question more specifically. So they actually, and I don't know how much, I'm going to ask my colleagues to kind of step in. I don't know how much they guide them on thinking of those things, but hopefully a lot of those things came up with the literature search. Kim?
Yeah, hi, good morning, everybody. Kim Kobajek here. I wasn't on yesterday because I was working, so welcome and thank you for tuning in today. When I did the CURE on forensic science with my students, I only had a few students, but they all had these very global, huge questions, and some of them just couldn't be answered. So I would do what most teachers do and ask them a question as an answer to their question about, can I do this? And I think, okay, well, just like Dr. Broach was saying, how are you going to answer this? What kind of data are you hoping to collect? And so I kind of did a little bit of both. I kind of let them go through the process and figure out, not too late because we only had 8 weeks to do this, and figure out, kind of finding out the mistake for themselves. But I'd also I'd made sure I give them guidance along the way, because there were a couple students in the groups that were not focused just on forensic science. They were focused more on the biology aspect of it. So I wanted to make sure everybody kind of got the learning experience, but of course not go off into the weeds and then figure out a week before their presentation is that they don't have anything to present.
Yeah. So the next question would be, how important is it for students to design their own experiment or to confirm their preliminary results? I mean, that was one of the goals of our CURE, was to allow them to take that extra step. And no, not everybody, a lot of people will just prescribe how to do something, but then they'll get out to the real world and and then they won't know how to design it themselves. So that actually was which is why we wanted to insert the and really insert the statistics students in the class, was so they do have that additional layer in our CUREs. I mean, that really was one of the one of the supplementary goal, like one of the aspects of the CURE that we wanted to do. And so so I really feel like it was very important to have the students actually do that. And the preliminary results, that's kind of the nature of the shortened CURE, and the fact that, these are student projects too. They may not ever come up with amazing data, but it's also, they're learning the process. Anybody else have anything to add?
Yeah.
Okay, I'll go to the next question. All right, and then so how long should we spend on this part and how much should we guide the students? So I think Kim Kobachak had already answered a little bit of that, but these are also available to the students as like kind of like worksheets or activities that they can even take home and do as a homework and then come back to class to discuss. So they're set up to kind of guide the students without much interaction from the instructor, at least at first, and then the instructor can come back and kind of help them modify it and help them make sure that they got to the right point. But it's not something that really requires absolutely tons of hand holding.
Wow.
Any other comments?
I agree with that, and that was something that I did. My iCURE was, like I said, during the summer. It was 8 weeks long. Students were there. They were great. They showed up 3 days a week, I think 3 or 4 hours a day. And that was a pretty tight schedule that we had to adhere to, but they were fantastic. So that was something that we introduced was, yeah, you're going to have this homework, and they would do it one night. They brought back their papers the next day, presented their questions. We'd kind of talk about them. If there was one that was especially challenging, I might take that student aside or that student group aside and say, you know, let's talk about what your question is here. So it was in pretty short order that we got things figured out, but also along the way when the students were performing the experiment, of course, stuff came up. So
yeah, I mean, that tends to just happen. I was going to address another question really quick. What suggestions do you have on helping students understand how to develop valid measures and measurements of concepts? And so the worksheet, the like activities, not only have an activity for them to see, like a blank activity for them to kind of like go through the process. There's also like filled in activities. So like they actually can go look at, you know, multiple examples of the cure, like of a cure example that was already done, just another generic like medical type example that was already done that had answers in there. So sometimes students learn better or like think about measurements as they see another example too. So not only are we try to guide them through choosing factors and choosing those things, but we also try to provide them a couple examples. And if once you've done it once before, maybe you can add in a previous student's examples as well, right? Like, so, I mean, once we've done it once, but at least there's a couple extra examples in there for their first time when so you can kind of see how somebody would develop from question to answer, to really testable question. Let me see if there's I don't see any other comments. Let me see. Oh, wait. What if a group of students really struggles with this process? Do you have any ideas of some bottlenecks regarding why they struggle? Maybe I'll ask my colleagues, how did you get a student that was really struggling with a question and or through that process?
I guess I reminded them that struggle is a part of science.
Yeah.
and let's say the experiment didn't go the way that they wanted it to or they thought it would. I always kind of say science happens, and you can insert whatever you want in there. And I did have a student group kind of struggle with not getting results, not even any results. And I said, okay, well, you still have results, right? Even negative results or no results are still results. This is still something to address. Maybe you have to go back and now kind of do some troubleshooting in terms of experimental design, or you just talk about this question in general. Do you think now that this can be answered? So I tried to guide him in a way where he still had a product, because he still did research, but it wasn't like he found the answers to this fantastic question that he had. So I just remind the students it's a learning experience, and if they want to continue with this, let's go back and troubleshoot, if you will, what could have been changed. And that's almost a part of their discussion and conclusion when they write their paper.
Thanks, Kim.
So we did have one more question, which was how long do people typically spend on this? And so I spent an entire class period on this. There was an additional question about how do you get them to do the experiments when you don't have the reagents? And I think we probably can talk about that after the challenges and successes, because I think each one of us sort of tackled that question differently, right? So in fact, this Q&A is, it's time to wrap this up, because it's time to move into our next video, which is called Successes and Challenges, where we all talk about what our greatest success and our biggest challenge are. And I think some of those will actually be answered. Some of the questions that are still in the chat will be answered. Any last comments? Okay, can we go into the video?
So iCURE is wonderful. It's a great example of how you get students outside of an academic or classroom setting and into a research lab. I think for a lot of them, that's exciting and it's scary, but you want to push those students to go in directions where they had never gone before. And I think that's why this program is so exciting.
The students typically were really, really into it. They spent a lot of time researching the topics that they wanted to study. They spent a lot of time forming their groups.
Students from different majors, putting them together to work in groups.
Only a couple of them were biology majors. We had everything in there from theater to psychology, you name it.
A lot of students don't like to be forced to work together. But I think when you do that, they see things from a different perspective. And the diversity aspect of that is so important, especially in our day and age.
Prep for a mini cure is very different from prep for a cure that's run for the entire semester, which can be 15 to 16 weeks.
Looking back on it, I was like, oh man, that was so much work and that was so much time. But during all of it, it was just a whirlwind where the students were so excited to do it that we just, got in and we just did it.
My students were really dedicated. They showed up 3 days a week for about 2.5, 3 hours a day during those 3 days to be able to get in all of their time to earn their credit hours.
We had 4 class periods to do this, which means a lot of the prep work had to be done well in advance before the students came to the lab.
If you're able to condense a research project that usually might take a semester, 2 semesters, into 8 weeks, but they did it.
One of the groups was so excited about their research that they themselves took their research paper and got it published in a student research journal. And that was solely motivated by them.
They get so excited, and I really think that's what it's all about, is getting them excited about science, excited about doing science, and being able to own their project, even if it is just for a short 4-week period that we're trying to incorporate into a larger course.
One of the important things about the iCURE is that it exposes students to a variety of different situations. And those different situations are not all successful. And I think that's good.
As we had one group that wanted to look at an abandoned farmland in South Phoenix. So you have both lead and arsenic in the soil. Well, this group that owned the land was trying to rejuvenate it. So apparently they put mulch on top of the land. And so when my students went out there to sample the soil, they did not find either lead or arsenic.
part of learning, part of science is also failure and how to respond to that failure. And working within a group, they can have support for each other and they can work through those failures and actually turn them into a success.
But what I then had them do is I said, well, okay, go to that farm site, look at the areas where we think the lead should have been, because it probably is just somebody put compost on top and we're using an instrument that's only sensitive to part per million. So I said, go out there and pick some of the weeds. Find the plants that are growing up on there, and then let's look in those and see what metal we can find. And in this way, they were actually able to show different levels of these metals in some of the plants on there.
I think an overall challenge is to try to get the students to simplify and instead of take this one big chunk, take these tiny little baby bites and start getting that big question broken down into smaller questions.
I had no idea on how to do a statistical test that actually looked at the length of the shoots and compared the two groups.
They had wanted to try something new, and it was an instrument. I had no idea how to do it. And I'm like, here's the instrument, here's the instructions.
We looked up the statistical test that would work, and they did it. And so this was a real success in that we all learned something that day.
I can tell you that if it had been the first or the second week of class and I had just handed that to the students and said, here, you figure it out, they wouldn't have been able to do it. But by the end of the class, the students just, oh, okay, here's a conductivity meter. We'll figure it out.
But this is one of the neat things about the class is it allows you then to have the students bring their skill sets in and use that to teach other students.
The kind of light bulb moment, the aha moment that went on in the students, I get it. I am a scientist. Man, I watched that video and I just get tingly in a good way because it really was just this amazing, fantastic experience. I wanted to address the one question, and maybe we can talk a little bit about it because this was a little bit of a struggle in terms of I can't find it now. It was about reagents. About the students designing their own experiments and then needing to order the reagents. And so the way I set up and scheduled my class was that there was one day where they finalized their finalized their standard operating procedures, and then they actually had to go online and they had to get me what I called a grocery list, a shopping list. Okay, so here's the question. Something I am struggling with is the openness of having the students design the experiments and then having the reagents available to test the question. What has your experience been with this? And so I actually had the students with their after they designed their experimental design and we went through it and designed their standard operating procedures, then they spent some time with the shopping list and what do they need, what do they want, and we had a very accommodating and helpful team that immediately turned around and was able to order the reagents and get them to us quickly. This was pre-COVID times. I know things are back ordered for 17,000 years at this point. And so if I had to do it again, I might try to constrain it a little bit more so that we have a very well stocked teaching supply area. And so try as much as possible to engage that because I mean, my favorite yellow tip is back ordered until November right now, which you probably think I'm a weirdo because I have a favorite yellow tip, but that's what it is. So that was how I dealt with that. Ken, how did you deal with the students wanting to order this, order that, or how did that work?
So the way I tried to deal with that is in the setup of the course itself, I have students, bring their proposals to the class for a peer review. But before that peer review, the students then, like what you did, is I have them give me a shopping list, and we figure out what's reasonable about it, and I go over it with them. Obviously some things we're not allowed to have. Even though my students did research on a uranium mine, they were not allowed to buy control uranium. That's how it is. But generally, it's working with the timing. It's working with your whoever does the support for your labs. And I think we're in a very fortunate age, at least before the plague, to have the kinds of overnight delivery services from those huge mega companies that we have that make this stuff a lot easier.
So I took a little bit of a different approach to this, in that I had a certain amount of equipment ordered, so sampling equipment, things to measure nectar. And so I let the students know what was available prior to them developing their experiments, because a lot of the equipment they would need is a little bit more expensive and takes longer to order. So I kind of constrained what they could do based on what I had available. So that's it.
And then I did a combination of what Dr. Marshall, Dr. Foltz-Sweat, and Dr. Sweat did, because I had students who, of course, wanted to run DNA, and getting a genetic analyzer like that is kind of hard, and of course it's going to be in the research space. And so again, shopping list, but also let them know ahead of time, some of these bigger cosmic questions like can we get measurable DNA from a fingerprint, may not be able to happen at this point in this iCURE.
So for my 4-week mini cure, I still wanted to give students options of what they could test, but we didn't have time to wait for deliveries. So I made sure to work with the folks in the teaching labs and use reagents that they had on hand. But I also encouraged students to look around for what was available locally. So what could we get from the local grocery store? What could we sample out from the environment to be able to get quick access to whatever it was that they want to study? For the larger Drosophila development and behavior cure that I did this past semester, I did a lot of what my colleagues did. I did have one kind of weird case where I had some students want to test fly behavior, but they needed this kind of elaborate contraption that was way too expensive for what we were trying to do in our course. So I encouraged them to think about how else they might be able to test it. And they actually took it upon themselves to go out to the local hardware store, find the materials that they need, and let me know, this is what we need and this is how we want to assemble it. So I encouraged them to be creative about how they can solve these issues if we can't order what they need.
That's great. Yeah, I think that each one of us had slightly different ways to approach this, but I think that that really worked. And the reagents that I was ordering, at the time were typical things that Fisher had on hand, and our Fisher orders are really pretty quick. And I know I don't own any Fisher stock. So we have a great question here, which is, what prep do we need to do on how to deal with failures?
I talk about it ahead of time, and I preface it that in forensic science, there's information, and you don't know the answer. That there are only a couple of parties that know the answer. One of them's not talking for one reason, and the other one's not talking for another reason. So I just bring it out up front that you may not get results. That's okay. That's the nature of science. That's the nature of doing these experiments. And it's not necessarily failure, because you do have results. You do learn something from it. So I try to put a more positive spin on it while also kind of giving them a heads up at the beginning.
So what I've done to address that, and of course this takes a while until you've had a few cures, but I have a large database of bee data. So like like Kim said, I talk to them about the fact that it's not necessarily a failure, and I have them evaluate their experimental design or their methods that they use. But in terms of data analysis, I can have them access this larger database that I have and ask a question that my students haven't necessarily asked before. And that will still give them data to do the analysis part.
So there's two strategies I took to tackle this issue. First off was pretty early on in the semester. As they were developing their protocols, I kept asking them, well, what if this doesn't work? And what if this doesn't work? So that they could start thinking about what the next steps would be. Because that's something we have to do in the lab all the time. You know, if this experiment shows something, then okay, we can go off and tackle that data. But if it doesn't work, then what are the next steps? What other types of things could we try? So I play the what if game a lot with my students early on. And towards the end of the semester, if things didn't work, I asked them to focus on what they did learn. And a lot of times that just goes back to really emphasizing the scientific method and understanding the process, and also understanding the fact that negative results, as my colleagues were saying, negative results are results. So they still learned a lot, even if they didn't get the results they were looking for.
So I agree with everything my colleagues do because I use a lot of those strategies. One of the other ones that I have come across because I've been this long in teaching that I find to be very effective is to salt the class with stories of your own failures, right? I tell the students I'm very proud of my work in organic chemistry because I consistently had about 110% yields on all my experiments, right? I talk to them about other things, and to be honest, I will sometimes take the mistakes my students have made. And as I tell the story to the next generation of students, they become my mistakes because I want them to see that I am making mistakes and it's not a bad thing. And so I think that kind of background strategy of humanizing it, really, it helps the students not only accept it, but it also helps them relate better to the instructor.
I think for me, the biggest thing with the students and the failure was that they were, the experiment didn't work quite right, or, there weren't significant results between the two variables. And I had to kind of remind them, you see what the grading scheme is, you've seen all the rubrics. Was there anything in any of the rubrics of the grading scheme that said that your grade was going to be dinged if your results weren't significant? And so I think for my students, a lot of their fear of failure was tied up in their worrying about their grade. And when we just reminded them, this is the syllabus, this is how we're grading, here are the rubrics for each activity, and you're earning an A, and so you will continue to, as long as you continue to perform the activities, that sort of alleviate a lot of those concerns. So there was a really good comment in here to go back to the shopping list, which was, we design experiments based on previous work in iCURE, so reagents are generally available. And I think that's a really good that's a really good strategy too, especially since there seems to be a lot of things that have been back ordered. It's another really interesting question. Has anyone had to deal with obtaining ethical approval before student studies could be conducted? And so what tips do you have for accomplishing that in a timely manner? And so Kim wants to chime in on this.
Yes, so my students wanted to use bodies to see how long saliva lasted on buried bodies. So thankfully, Dr. Marshall was also working during the summer. So I'm asking her, we can't get dead bodies. What do you suggest? So we were looking at the frozen rats. And this was all happening within a couple of days. And so finding out what the students wanted to do ahead of time was very helpful, because then I could start calling and asking people, because I did need to contact IRB, and instead of just the institutional review board. So instead of just relying on emails back and forth, I am getting people's numbers and I'm calling them. And I had all of that information ahead of time. And again, thank you to Dr. Marshall for that. How were these animals euthanized? What are they being used for? And because even, I don't know if this is an urban myth or not, but I guess even if you go get chicken or beef from the butcher, that could be an issue. So I had this question and I got an answer in, honestly, a fairly short time. I'd say about 2-3 days. I had an answer that yes, I could use these particular specimens. You don't need IRB approval. And when I had talked to the representative from IRB, they said, just if we have to get this a quick turnaround, just make sure all your I's are dotted, all your T's are crossed, complete SOPs, and we'll try to, expedite the process.
I just wanted to quickly add, I appreciate you asking that question, because we should be integrating a lot of ethical concerns throughout any of the cures, because even, sometimes I think we think of, the dead bodies needing some other protocol. I mean, you could tell in the statistician that doesn't even know what half of these reagents are. So at any rate, but there's also ethical concerns and like social science questions. And you can't just go out and ask your classmates things at the university, because that's just not okay. Like, so certain things that they think, students might think that, they might have to ask permission to get a dead body. They also need to ask permission for a lot of things. And so, and there's so many, I work with so many socials. And a lot of you guys will do social science type experiments, and the ethical concerns about those are different but equally valid. And also we can't ask people about their grades, we can't ask people about their socioeconomic status, we can't just send out a survey to the classroom. So they need to know that. They need to know how we can ethically and and follow the rules, right? And follow the guidelines that are set forth. So I think sometimes it's super obvious in Kim's situation, but not so much when they think they can just ask all their friends, because then we are at the university and we are bound. We have to protect our students.
So if I could add to that, yes, it's very important you have these kind of ethical, these ideas in your cure. Ethics in science is really important. And it's important students understand that as well. All of the things that Dr. Broach was just talking about in terms of what information we can get from students are very important for them to understand and realize. My advice to you is, if you foresee this happening, if you foresee that you would need institutional approval for a cure, is to start working on it as soon as you can. And to start, even before the semester you're teaching it, start building those relationships with your institution's IRB. And also, what I would recommend for like the social scientists that are here is a mini cure that's focused on the IRB process, right? So yes, you have a research question, but you could actually use that as a mini cure in a social class to teach students about the IRB process, about how you can ethically harvest data from human beings. But as a final note, this is also why I work with plants.
So I did want to follow up. We, when we were awarded the grant from the National Science Foundation, before we were able to activate the grant, we had to have our IRBs in place. And so we do have IRBs in place because we are asking and answering questions about students' trajectories, students' careers, and students' ability to complete a degree with and without the iCURE, as well as some of their demographics. So we do have that in place. ASU has a very robust infrastructure for animal care and use, human subjects, IRB, as well as recombinant DNA. And so we have many, many websites and many, many trainings that we have to take in order to really be able to, do the experimentation that we need to do. And I see something in the chat that says, I definitely see the merit in working on the ethical approval for fall now. And I wanted to share, not with the iCURE, but I had an honor student who wanted to do her thesis. And I walked her through it, and she wanted human saliva samples to do some microbiome. And I told her, sure, but you have to take city approval. You have to write the protocols. You have to write the IRB, and I will submit the IRB. And she was really taken aback. And it was a lot of work for her. And I helped her. I didn't, send her adrift. And then at the end of it all, I said, I did that on purpose. It was very, very difficult, but now you have something that you can use for your medical school application that sets you apart and above and beyond everyone else. And so I think at the time it was all happening, there were words that she was saying that she didn't want me to hear. But at the end of it, she actually thanked me, So sometimes work is hard. Let's see. So we just completed 2 semesters of analyzing the nasal microbiome of students who tested positive and negative for SARS carriage, and we walked students through the IRB process. Yeah, I bet that was a lot of work, and I bet while the students were doing it, it was complicated, but I bet they learned a ton. And I tell the students, I said, you just took a bunch of safety classes. This is for our summer program. Those should go on your resume. Like, oh yeah, that's a good idea. So try to, talk up the positives. You've now done something that people you're competing for haven't done. And we did have something in the chat that said, if you're meeting the student learning objectives, but the students don't have papers, then they're at a disadvantage for jobs. I can see that, but if the students play up what they have learned and what they can do independently, I think that is a real positive as well, because there's a lot of things that I do in my research lab that I haven't gotten papers for. Let's see. So we got another question that actually is a really good lead into our videos that will be coming in a few minutes. It is, does iCURE include ecology or environmental questions that do not require many lab supplies?
So I can go ahead and speak to that. So my pollination biology cure is obviously environmental, and it's usually field work. It doesn't require a lot of supplies every semester, but there is an initial investment in equipment. Like, for instance, I have equipment to measure sugar content in nectar. I have the little capillary tubes, nets, pan traps, all of those things, but I can reuse these every semester. But absolutely, you can use iCUREs to ask environmental or ecology-based questions, and I highly recommend that, being focused on pollinators.
The other thing is James Filikowski, the one that talked initially on the Successes and Challenges video. His research is on sharks, and we did not send all of the students out to the ocean to to go study sharks. So they were all analyzing very unique research questions, but from previously collected data, and that's okay. That's fine. And there's also, so they did a whole cure on previously collected data. They still have to do the same things. They just don't go through the physical steps of collecting the data, but that doesn't make their cure any less scientific, or that whole it doesn't change much of the research process. And it could also help you even speed it up from a social science perspective as well. Like, there are a number of databases out there that are national databases, crime victimization surveys, there's lots of really cool questions you can ask. I know one of the cures that we partner with Phoenix College, their mini cure, their cures are embedded in already existing courses. They're not full courses. They partnered with the crime lab and the Phoenix Police Department, and they're looking at crime trends and things like that. So they were given a database of, like a huge database, but they weren't given specific research questions. They knew a methodology that they were able to do, but then they were able to ask different questions of the data. So that's always an option, and that doesn't require any tools, because there are freely available tools for analysis online. So that's one thing that you can do, and that just changes. And that also could be for ecology. I know, like, the IUCE and Red List has all of their data freely available. There's lots of available NOAA. There's lots of freely available websites that we had kind of posted yesterday, a few of them during the breakout session, that you can use to do environmental studies or ecology studies. Anyone else?
So yeah, field studies that require relatively little resources, I think actually are an incredible way to bring cures in. You'll notice in some of my phytoremediation videos, I talked about running transects and doing plant sampling. That can be done with just a transect. And in fact, if you dive into the literature on, I'm gonna go a little bit away from plants here. If you dive into the literature on lichens, which is where I did my dissertation research on, lichens are known to be highly sensitive to air pollution. And all of that research was worked out by research studies of literally transects and measuring populations at distances from pollution sources. So there's an incredible body of research already out there about doing these kinds of field studies that really only require you to be able to quantify the numbers of the organisms you're interested in. So I would highly encourage one of those field courses. And yes, it should be really easy to do so with a minimal support, resources, as long as you couch your questions appropriately.
Okay, great. I have put several different links for several different databases here in the chat. So hopefully that gives some people some ideas. Let's see. I think we answered all the questions in the chat. Are there any last questions? We have a few more minutes until our next section. Any last questions? Oh, we posted that twice. Okay. All right. So I don't see any last questions. So I wanted to let you know that our next section is all about using bees as a study model. And these videos are from Dr. Foltz Sweat. So I should probably throw in another Shakespeare joke here about two bees or not two bees, right? And interestingly, I never knew that there were any kind of bees besides honeybees. And Ken and I were out looking at some rosemary, and he pointed out to me, hey, look, that rosemary's got a bee on it, and it's not a honeybee. And I looked at it and I was like, that bee is blue. Bees come in blue? So Jennifer Foltz-Sweat, Dr. Foltz-Sweat, Ken is teaching me bee biology. So you've done something right.
My name is Dr. Jennifer Foltz-Sweat, and I've run several cures at ASU. I want to take a minute and talk about the different types of projects and questions that my students have asked over the semesters, having to do with bees and pollination. So during one of the first cures, one of my students looked at the difference in bee species richness and abundance in a semi-natural habitat, Piestewa Peak, and an urban area, the area surrounding the ASU campus. So he surveyed bees and he extended past the cure, looking at these differences. And also you can look at whether bee species richness and abundance differs between spring and fall, or from year to year. So it can be an ongoing project with your CURE classes. Another project my CURE students looked at is whether bees prefer different flower types. So you have the tubular flowers and then you have flat flowers. And so this student or group of students used observational sampling of different flower types to look at the variety of bees that come to these flowers and also the abundance of bees that come to different flowers. Now, I've also had students measure nectar in the flowers and look at whether that influenced bee foraging. So you want to look at the questions or the project that your CURE students will be working on, and then you can determine which type of sampling method to use. So when you use what we call netting, targeted netting, you are collecting bee specimens and you are documenting their floral associations. Now, the caveat to that is this is very time intensive, both collecting and grouping the bees. You can do morphospecies, which means that you put them in different groups that look alike. So you can say, I have so many of this bee, so many of this bee. Now, pan trapping gives you an idea of abundance, a little bit about species richness, although it catches certain species more than others, but you have no floral association. So students can use pan trapping to look at the overall abundance of bees and maybe a little bit of variety in different habitats. Observational sampling, students are not collecting specimens. They are looking at how many bees and the different types of bees come to flowers. This is the method I prefer for cures because it is less time intensive and students do not have to try to identify the bees down to a species or morphospecies level. So just to give you an idea, when I identify bee species, it can easily take 30 to 45 minutes for me to identify one specimen if it's something I haven't seen before. So again, that netting and pan trapping are very time intensive methods. So now we will go on and look at these different sampling methods and how you process bee specimens. So I am going to show you how to prepare a kill jar, which you will use if you are going to net the bees. So this is what a kill jar looks like. On the top, it says BioQuip, and you see the poison symbol, and that is because you are going to put a chemical called ethyl acetate into the kill jar. Now you want to do this in the fume hood. So here I have the fume hood, I have my gloves on. I'm going to set the jar in here. Now this is plaster of Paris. You're going to unscrew this. And I put cotton in the top of the kill jar. And what I'm going to do is add ethyl acetate until the cotton is saturated. In fact, you want to see that it is soaking into the plaster. That's how you know that your kill jar is ready. Now, some people sniff it. I don't recommend it. It smells like nail polish remover. So in the fume hood, I will just pour the ethyl acetate into the lid slowly until it is what we call charged. Screw it back into the cap. I generally, like I said before, put paper towel, and your kill jar is ready. So now that we have prepped the kill jar, let's go outside and sample some bees. So we talked about cures, a bee cure, and the types of questions that you can ask. So now I want to show you some netting techniques. So here are two types of nets. This is what I call a student net. These are actually a lot cheaper, inexpensive. This is a professional net, which costs a little bit more. They range around $100. So here is the benefit of this type of net. If you look close at the mesh, the student net has larger mesh. This net is really tightly woven. So the difference here is you can catch very small bees with this net as opposed to medium and larger size bees that you'll get with this one. Now I use the professional net, but if you're running a cure, you may want to go for the other type of net, especially because these really small bees are hard to see. So now let's go and do some netting on this patch of mallow, the scientific name being Straussea ambigua. As I walk around the patch, I want you to notice that I walk in a way so that my shadow does not fall on the plant. If a bee is foraging and a shadow comes over, it will generally fly away. The other thing I want to point out is that you should sample when it's sunny out, very little cloud cover, above 70 degrees Fahrenheit, and not very windy. Okay, so let's get started. So the first thing I'm going to do with this patch of flowers is walk around the perimeter. Now remember, you need to walk so that your shadow is not falling over the plant. I want to stay about 2 feet away from the plant so that I don't scare the bees. Now I have the honeybee in my net. What you want to do at this point, bees will always fly up. So you want to keep your hand right here. The bee is right here. Now make sure that the net does not come over your hand enough so that the bee can sting you. So in my pocket, and this is where I usually carry my field equipment, you can also get a pack to put around your waist, but this is what we call a kill jar. And obviously I use it to kill the bees. Now I want to make a note here. Native bee populations are declining. So I do this in a very cautious way. I don't want to catch too many bees. We have a honeybee here, which is from Europe, and these are not declining. Here's the top of my kill jar. Now it has ethyl acetate, which is kind of like nail polish remover. So what I'm going to do, I'm going to hold the lid in my right hand, and I'm right-handed. I'm going to feed the jar up through the net, making sure not to let my bee go. So I keep feeding the net through my hand until the bee is in the very top of the net. It flies into my jar. I put the lid on over the net. Now I'm going to hold it here for about 30 seconds until the bee stops flying. And you want to take it out of the net like this so you can watch the action of your bee. You don't want it to fly out of the jar. Now, see, my bee is starting to slow down, so I can remove the cap from the inside of the net, bring it out, seal it. Now, an important note about this, one of the objectives of targeted netting is that you can make an association between the bee, the type of bee you caught, and the plant that it is foraging on. So depending on your students' research questions, you may or may not want to use this technique. But since I caught this honeybee on this desert mallow, which is Straussii ambigua, I am going to make a label so that I remember where I caught this bee. Now, as I'm sampling this patch, I can put multiple bees in my kill jar, and it doesn't matter if they're the same kind of bee. They could be all different species because I'm going to put them in different storage vials with different labels. So for this sampling method, I have a volunteer, my husband, Ken Sweat. He is going to show you how to do this actual technique. So what I want to show you is pan trap sampling. It's also called bee bowls. There are a few things you need to know before you start this technique. So first of all, I use these solo plastic bowls. They come in white, but you need white, fluorescent blue, and fluorescent yellow. So I always buy spray paint at like Ace Hardware, that's fluorescent, and you paint the inside of your bowls. Now keep in mind that if you're in a region like Arizona, you're going to need to redo your bowls halfway through the season because they crack, peel, and break. So we are going to lay these pan traps out every 5 meters, and you'll do this on a 150 meter transect if you can. This is just the standardized protocol, but you can alter it as needed. So you will be using 30 pans, and you need to do it in a color sequence. So for today, I have yellow, white, blue, so it will go yellow, white, blue, yellow, white, blue, okay? Now, in your pan traps, you are going to put water with Dawn blue dish soap. Now, again, standardized protocol, this is what we all use. And the reason is kind of funny. We use it because we just look at the color of the water to determine when we have enough soap. I usually put approximately a tablespoon of Dawn per gallon of water. But let's squirt some in here. You want it to be a light blue after you mix it up. Maybe a little bit more. I think that looks good. Now, this is a surfactant so that the bees, once they land in the water, they can't fly out of the water. So again, this is a technique where you are actually killing the bees and bringing them to the lab. The good thing about pan trap sampling is that it's unbiased, meaning it doesn't matter who's putting out the pans, you will catch the same types of bees. Whereas netting, it varies a lot depending on who is doing the netting. Now, one con about using pan traps is that they tend to only catch small to medium bees. The large bees do not come down to the pans. And by large, I mean like bumblebees or carpenter bees. So let's get started. So today we are going to put down 3 pans, but remember you will be using 30 if you're on a 150 meter transect. Now, for those of you who are wondering what a transect is, it's just a straight line that you measure that goes through whatever area you are trying to sample. So for bee sampling, I like to put my transects in the middle of the area, but you can also do an X formation so that you are getting more area. I generally do 2 transects per region, per habitat. So I'm going to have Ken place the pan traps, and then he will go back and fill them. You fill them about 2/3 of the way. And again, this kind of depends on region. In Arizona, a lot of my water in the pan traps evaporates throughout the day. I generally place my pan traps in the morning before 9 a.m. and I pick them up around dusk, which is about 5 o'clock.
But again, you have to make sure, like in some areas that I've sampled, the wind starts to blow around 3 or 4 o'clock, and that will blow your pan traps over. So just make sure you have a protocol that matches the particular area that you're sampling. Here are 3 pan traps, and like I said, I decided to do yellow, white, blue. It doesn't matter which order you put them in as long as you're consistent. So he starts here with the first pan. Now, notice there are a lot of soap bubbles in this pan. You do not want that. Bees will see the soap bubbles and they will not land on this. The idea is that they think this is a flower. So what you want to do is pick up your pan trap, and I lightly blow on the bubbles so that they are blown out of the pan. Now, granted, they will go down after a while, but you want to give the maximum amount of time for bees to see your pans. That looks better. Now, Ken is going to start at this spot, and he will pace out 5 meters to the next pan. Wide step, or 1 meter is a wide step. And so Ken is going to remove the soap bubbles. Very good. Now, one other thing I want to point out, and Ken will demonstrate this with the next pan, you should be going through vegetation. You don't have to go on a clear path. And if you're putting your pan in vegetation, you want to make sure it's flat, and you want to clear the vegetation away from the pan so that the bees can see it and access it. Now remember, you are filling these pans 2/3 full. You can fill them more, but if you're out in a semi-natural area, that's a lot of water to carry with you. Now, I would generally carry, if I'm laying 2 transects, 2 of those full jugs. So at this point, you have left your pan traps out for the entire day. So now I'm going to show you how to collect your bees from the pan traps. So here I have what we call a whirl pack. Now these work best for collecting your bees. All of the items I've shown you so far, you can purchase from an insect supply company such as BioQuip. Here we have a strainer, which is how you will get the bees out of the water. Now, one thing I want to mention on your whirl pack, make sure to put the date and location that you sampled in, because if you don't do it right when you're collecting, you will forget that information later. Now, Ken is going to collect this honeybee that flew into our yellow pan. So you pour the water through the strainer so that your bee ends up in the strainer. Now, for the whirl pack, the top comes off. What I usually do, there are tabs on the side to open it, and then I give it a quick burst of air so that you can put the bees inside. This is a difficult part. I have tried various techniques to get the bees inside the whirl pack. A lot of times you have a lot of small bees. I've used like a spoon to scoop them out, whatever works for you. Now, after your bees are in the whirl pack, you are going to use ethanol. And this is not rubbing alcohol. It's an ethanol that is at 70%. You're going to use this to store your bees. Now, you don't need to fill it to the top. Just fill it to a reasonable level depending on how many bees you have. Now, to seal the whirl pack, this is important, otherwise all your ethanol is going to run out. You want to squeeze it shut like this, take the air out, and now you are going to fold it down. I'm going to fold it down pretty far because I don't have much ethanol. And then use the twist ties, and you just fold them in. That is how you want to store your bees that you catch from pan traps. So now I'm going to show you the last sampling method that you can use. And this is actually one I prefer for cures because you are not taking a lot of bees out of nature or in an urban area. So I'm going to go over observational sampling next. So we're back at our patch of Straussea ambigua, or globe or desert mallow. And what I'm going to show you now is observational sampling. Now keep in mind, with netting and pan traps, since you're actually collecting the bees, you can identify them much more specifically. In fact, with students, I use what's called morphospecies, where you separate bees depending on how they look. Now, observational sampling, you are going to look at bees and put them into broad categories, like large bees, medium bee, small bee, also maybe based on coloration. These are categories that you can define depending on your research question. So I have my field notebook, and I actually use a micron pen because it's archival quality. It won't fade over the years. Here's my field notebook, and I'm going to do an observation on this plant. Now, when you do observational sampling, you walk around, you find a particular plant with a lot of bee activity. Now, what you're going to do is look at the plant about a meter squared area. It doesn't matter if you're exact, but that just gives you an idea. So for this particular plant, I would actually look at most of the plant. You're going to time yourself, and you will observe bees for a minute. Now, if you look at this plant, you see honeybees going into the flower, collecting pollen and nectar. You want to look at bees that are actually inside the flower or on the flower foraging. You don't want to look at bees that are just flying in your field of view. So I would stand here for a minute. I see 1 honeybee, 2 honeybees, 3, 4. So you're just going to keep your eye on the plant, and you would have your broad categories on your field notebook, and you just make marks. I would make 4 lines for honeybees. Now, in order to repeat the process, you want to do 3-minute-long observations on this plant. And that ensures that there's some consistency between your observations. So I showed you how to do 3 sampling techniques: netting, pan trapping, and observational sampling. Now I have some bee specimens here that I obtained through netting and pan trapping. Remember, for observational sampling, you will not be collecting any specimens. So I'm going to show you how to prepare your specimens for curation or mounting. So here are the bees that I have in my kill jar that I got through netting. These are the easiest to prepare for curation. I take off the lid. Now I had a paper towel in there because it helps give the bees something to hold on to. And in here I have 3 bees and leaves. Now what you want to do with these bees is put them in a storage vial. If you're going to mount them immediately, you don't need to do this. But when you collect bees, they can stay in your freezer for about a week. If you leave them longer than a week, you will have to relax them, which is a whole different process. So here I have a storage vial. You can also get cheaper and smaller storage vials, but these are the ones I like. Now, if you recall in our previous video, I told you that you have to make a label when you collect your bees. So on my label, I have written the date, the location, I wrote which transect I was on, and the name of the plants, Straussea ambigua. So I'm going to keep all of the bees that I got from that area together. Actually, you can just pick the specimens up with your fingers. They are very pliable at this point. If you freeze them for a week, they may be a little more brittle, so be careful. Now we will go over how to prep bees that you've collected in ethanol. Remember, this is your whirl pack, so I'm going to go over to the sink. This is what we like to call our bee salon. So you're going to open the whirl pack, Remember, ethanol is not going to hurt you. You can do this with gloves if you like, but I tend to not use gloves for it. You're going to pour the ethanol into your strainer. Now you have to rinse your bee. Remember that they are pretty pliable at this point. So you just run a stream of water over your bee. Now, even if you're looking at pollen on your bee, it will not come off while rinsing. So now that you have rinsed your bee, you are going to do the first part of drying and fluffing your bee. Remember, you want the hair on the bee to show. So we're going to put it on the paper towel, and I have 3 paper towels here. You will put 2 paper towels on top, and then I pat the bee, and you can see water is coming off the bee. So the reason that you need to dry and fluff your bee is because we use the hair on the bee, and we call these pollen collecting hairs or scopy, to identify the bees, among other characteristics. But you need your bee nice and fluffy in order to identify it. So I would leave my bee sitting here for at least 15 minutes until you start the blow drying process. Now this is the fun part. Here I have a mason jar, obviously. Now I have taken a very thin sock and put it over the opening of the mason jar. You can also use nylons if you can still find them. I know these aren't really in fashion anymore. Now what you're going to do is put your bee or bees in the mason jar. You want to keep all your bees from your transect together, but don't mix them from multiple transects. I'm going to put the lid on. Now, you want a blow dryer that you can set to cool. You do not want to blow dry these bees on warm or hot. You will make them too brittle for mounting. So I have the bee in the mason jar. I'm going to turn it upside down. My blow dryer is on cool. I usually put it on high power, and I blow dry like this. I turn it upside down so that I can blow the bees off of this surface. They have a tendency to stick to it. You want the bees to tumble around in the mason jar and become nice and fluffy. So you can look at your bees after you blow dry, and I usually blow dry for maybe 30 seconds at first, especially if you have small bees. Your small bees will get brittle very quickly. In fact, you can even remove your small bees after like 30 seconds and continue to blow dry your larger bees. So now I take my bee out, I look, and I see that the scopy are fluffy. So now you are done preparing your specimen. So now that our bee has been processed, we are ready to go to the microscope for curation, which means mounting the bee. So now we are ready to curate and mount the bees that we collected. So there are a few items that you need to lay out before you begin mounting the bees. So one of the most important items is the pin or the pins that you are going to use. So you only want to use number 2 insect pins. They come in a variety of sizes, but number 2 is what we always use for bees. They come in these packs, and they come in different directions. So some are facing down, some are facing up. So what I do is sort them all so they are in the same direction and put them in a vial. You will need a mounting block. Now this is one of the higher end mounting blocks that I use. However, these are the mounting blocks that I have the students use. These are relatively inexpensive. And the mounting block or pinning block is used so that you put all of your bees at the same height. When you are mounting, attention to detail is very important, especially if you are going to try to identify the bees. Now I have a cork, and I like this one because it stands really well. Once you have your bee mounted, you can use this cork to look at it under the microscope. Finally, I have 2 types of forceps. A lot of people call these tweezers, but we call them forceps. I have what are called featherweight forceps. They're very light, so you're not going to crush anything that you pick up with these. And then I have these fine forceps. Now these are what I use to spread the wings and straighten out the legs. If you drop these, they will not work anymore. Once they're bent at the tip, they're not very useful. So now we're going to mount the bee. This is a honeybee, a medium-sized bee. And when you first learn how to curate, I recommend you use honeybees. Now I'm going to take a number 2 pin and you are going to pin the bee in the thorax. Now remember, a bee has 3 segments: head, thorax, abdomen. The thorax is the area where the wings attach. Now on the screen here, you can see that there is a center line on the thorax. You want to pin the bee just to the right of that center line. For certain bee species, we use the center line to identify the actual species, so you don't want your pin to go through it. Now here I have a foam pad, has card or wood underneath. This is what I use for pinning. Some people use different things. If you don't want to purchase the foam pad, you can use just styrofoam that you get in a package. That works as well. I like to put the bee on the edge of the styrofoam. Now it's important that your bee is straight on the pin. So I usually hold the bee between my thumb and forefinger. Now I am going to mount or put the pin through just to the right, and I'm obscuring your view momentarily. Usually I will put the bee on the pin just a little bit up the pin so that I can look at the front of it and make sure it is straight. This one is pretty straight. It's tilted to the side a little, but once you mount it, you don't want to remount it because you will make a large hole in the thorax. So now that I have it pinned, I am going to use the mounting block. Notice that there are holes at different levels. That indicates how far down your pin will go. So see, there are holes on the top of your mounting block, and that is what you use. So I'm going to use the mounting block. I will actually use this deep hole right here, and I'm going to push the pin into it. Now you can see that my bee has been pushed to the top of the pin. But now I'm going to use this hole on the end. I'm going to turn my pin upside down and push it that way. The reason that I do that is that different size bees will go to different areas on the pin. So I want to turn it over to make sure that this area is the same for all of my mounts. And you want to do that also so you have enough room to grasp the pin. After you have pinned your bee, you can always go back under the microscope. You want to make sure that the wings are spread out. Now remember, bees have a forewing and a hind wing. They have 4 wings total. And that's actually what you can use to identify bees as opposed to flies. There are many flies that look like bees, but flies have 2 wings. Bees have 4. We use the veins, the wing venation, to identify bees as well. So you want to make those wings visible for when you go back for identification. You also want the legs to stretch down as much as possible. When a bee goes through a kill jar or is collected from a pan trap, a lot of times the legs fold up under the bee, and you can see that here. So you want to stretch the legs out. Now make sure you do this carefully or you will pull the leg off. Now I generally use the fine forceps, the ones with the point, to position my bee and to spread the wings and make the legs come down. So now we will be looking at bee identification. So now we are moving on to identification. Now you can have your students collect bees in the CURE like I mentioned before, or you can do observational. Now this is showing you bees that I have collected from a study. The first thing I want to point out is there are two different types of boxes that you can use to store your bees. This is what I use for students, and this is a very cheap box. It has styrofoam on the inside, cardboard for the bottom, and the lid. Now, the problem with this type of bee box is you will get tiny beetles called dermestids that go in and eat your specimens. So you will want to put these in the freezer or circulate them into the freezer every month or so. This is the box that I normally use. Now, these boxes are about $50, so they're probably not feasible for cures, but they are very good at preventing dermestids from getting into your box. Now, the other thing you can do when you use a student box is you can buy strips with chemical on it that deters the beetles. It's a little bit toxic, so it's up to you. Now, what I want to go over with identification is some of the main characteristics that you can use to group bees. I'm not going to go into the finer points of identification because there are many, many characteristics we look at in a dichotomous key. So let's go over here. Now, one thing I want to show you, this is a pamphlet that I made for a citizen science project in California. And what I want to show you is some of the information that you can prepare for your students to help them group bees in the field, especially if they're doing observational sampling. So here we have a carpenter bee, and I am showing you a picture of a female and a male. Now, under here is some basic information about carpenter bees, and I wrote down floral hosts, the plants that we most commonly find carpenter bees foraging on. So this would be a very useful item for identification, and you can make it into a PDF so that your students have it on their phone. So I want to show you some of the diversity that you will see, especially with Arizona bees. Keep in mind that we have approximately 1,000 bee species in the Sonoran Desert. So this is a variety that I pulled out here to give you an idea of the variation that you'll see. So the first bee that we have here obviously is metallic blue, and you probably have never seen a bee that has this color. These are pretty common bees, and they generally come out in the spring. We have certain bee species in the spring, different bee species in the fall. This bee has a very distinct striped abdomen. This bee is called Anthidium, and they are fairly common in Arizona. I pulled out this bee because this is the most common bee you will see here. This is a sweat bee. Now, that is a general categorization. These bees nest aggregately, so if you pan trap, you may get a lot of these bees in your pans. This bee I pulled out because it's one of our smaller species. Now, in Arizona, we have the smallest bee species here, Perdita, and one of the largest being the carpenter bee. And finally, here is what we call our leafcutter bee. Now let's go over some of the characteristics here that you can use to group bees. This leafcutter bee, the blue bee on the end, and the one that I have in the microscope here, all have scopy or pollen collecting hairs under their abdomen. That is a very key characteristic to this group. This group is called Megachilidae. Now that's a family name. I just call them megachilids. So you can actually make that one of your groups that students put bees into. It's a little harder to see the scopy in the field, but if you look carefully, you should be able to see pollen on it. Now, another grouping would be, of course, small bees, and then you could have medium bees being like your honeybee or your Anthidium. Other characteristics that I look at is where the bee has their pollen. Now, like I said, the megachilids will have it under the abdomen, But if you take a look at a honeybee, they have almost a blade-like hind leg, and the pollen is formed into a ball, and it is attached to the pollen hairs on the blade-like leg. So if you are ever curious about whether it's a honeybee or a different type of bee, just look at where the pollen is. Honeybees are one of the few bees around here that have the pollen in a ball. Other characteristics you can use to group your bees, of course, would be color. We have black bees, brown bees, metallic bees, bees with striped abdomens. This way, you can get a really broad idea of what type of bees you are seeing in the field. And if students are doing observational sampling, you can also get an idea of relative abundance. Like, are you seeing just a few metallic bees or a lot of metallic bees? Now, with observational sampling and netting, you can always make a floral host association, and that is very valuable information. But it also depends on the questions that your students are asking. So that concludes identification of bees. And I want to reiterate that when you look at the types of questions that your students are going to be asking, you will decide on the sampling method. And if it is observational, maybe your students can go out and decide on the groupings that they may use.
Thanks, Jennifer Foltz-Sweat. And the reason why I use last names is because we have 3 Jennifers. And so we're considering all changing our name to Jennifer. Well, and a Kennifer. All right, so I learned a lot actually from that. I didn't realize that bees had 4 wings and Drosophila had 2, or flies had 2. So I learned quite a bit. All right, so it is the question and answer time. So we did have one person asking to post the PDF of the Citizen Science Bee Identification doc, and so we are going to post that. That is for California, and
Right.
we will post that as soon as we are able. So additional questions? All right, well, we're waiting for questions to come in. I remember that there were some timing issues that occurred with your cure and the bees.
Yes, this is actually a big difficulty with the pollination biology cure, is that I will lay out the timeline for the course, but it's dependent on when the bees come out, right? They're all nesting in the ground or in cavities in wood, and when they emerge is highly dependent on temperature and precipitation, because they want to emerge at the same time as the floral hosts bloom. So this has always been an issue with my cure, and one thing I do about this is I try to keep it very flexible. So I have alternative exercises prepared just in case we have to delay data collection.
So we had a question from Tess. How often do you get stung?
So that's a funny question. So yeah, I personally I don't get stung, but I have only had one student get stung while collecting bees, and he just wasn't paying attention to where the bee was in the net. But I make sure, first of all, I make sure that none of my students have allergies to bees, and if they do, that they have an EpiPen with them. But generally, I go over netting techniques pretty thoroughly so that my students do not get stung. And I also have them do netting the first time with me there during lab.
Yeah, the only time I ever got stung recently was when I was trying to save a poor little bee out of my swimming pool, and she didn't quite realize what I was doing. And so it was just bad for us all. So we have another question here. Do you make the students use dichotomous keys during the lab to identify bees?
So that's a really good question. I have dichotomous keys available, but it is, if you've used a dichotomous key for bees, it's very difficult for students to get past genus, of course, because the key that's available for the Southwest only goes to genera or genus. And so in order to go to species, I have to find monographs. But in general, it's just very difficult to use the dichotomous key for bees. So what I do is have students divide up their specimens into groups that they think look the same. So we go to morphospecies. I talk about the different characteristics that they should look at and how they should group the bees, like placement of the scopi, which are the pollen collecting hairs, coloration, size, things like that. So it is not it's not ideal depending on your question, but if your question is just looking for a relative abundance and a basic idea of bee species richness, then morphospecies is sufficient.
So one of the other questions that came up in the discussion, Jen, is with pan trapping, do you look at wind forecasts to determine when to pick them up? And is there a way to weight down those pan traps?
So that's a really good question. Yes, I do look at wind forecasts, but as a general rule, our afternoons tend to be windy, so I kind of count on that. It also depends on the area that you are laying the pan traps. If it's really bare with not a lot of vegetation, then yes, they are going to blow over. So I have looked at a variety of ways to weight down my pan traps. I put a few rocks in the bottom to weight down the pan traps, but you have to make sure that the bees cannot stand on the rocks. Obviously, then they don't fully submerge into your pan trap. So that's one of the main methods that I've found. I'm looking at the other questions here. Okay. I don't see any questions that I haven't addressed.
Sorry, I had a technological glitch there. Can you talk a little bit about the student paper in the Journal of the Arizona-Nevada Academy of Science?
Yes. So with the student paper, the work began during one of my cures, but this particular student stayed on and worked with me for quite a few semesters. So he collected very useful data during the cure, looking at a comparison between a semi-natural habitat, so several locations in the Piestewa Peak area, and then several locations in the urban area around ASU West. And so he, based on the data that he collected during the cure, he collected actually quite a bit of data. We decided to pursue this research question further. And one point about that is a lot of times students get so interested in the projects that they do want to continue on with independent research. So we ended up publishing our data in the Journal of the Nevada Academy of Sciences. So this is a really useful journal that focuses on a lot of ecology studies, and so we thought that was an appropriate place to publish.
Yeah, I've published student projects there as well. It's the Journal of the Arizona-Nevada Academy of Science. Actually, I've been told that's not how you pronounce Nevada, it's Nevada. So how did that work when you published? How much were the students actually able to write the paper?
That's a really good question. So this particular student, we worked for probably 6 months on the paper, and he did most of the writing and analysis of the data. Now, this particular student was very far along in his academic program. He was a senior and very motivated. So he used the writing and publication of the paper in his medical school interviews, which were successful. He did go on to medical school. But it was a great deal of work, but I did let him take the lead on writing, and I actually let him first author the paper. But, you know, it depends on the contribution of the student.
All right, so we had another question here about the replicates. As for replicates, how many times do you recommend the students to collect the data?
So that's a good question. In the study that I did, or that my students did in the semi-natural area comparing to the urban area, they were netting and pan trapping. I believe he sampled every 2 weeks in spring and fall. Now that's pretty intensive netting of bees. I probably would not do that again, given the further decline in bee populations. Now for observational studies, for a full cure, again, I recommend that my students sample every 2 weeks. So they usually get 4 or 5 replicates. And in the mini cure, I'm going to ask that my students sample 3 times a week, which I know Jennifer Broach would kill me for, but given the limited amount of time that we have to collect data, they're going to do about 3 replicates. And if they have more time, depending on when the bees emerge, we'll add replicates.
So let's talk about this 3. 3 has always been, you know, well, Excel won't let you really do a t-test unless you have 3. I know you're laughing. But when you don't have a lot of time and maybe you are a little bit underpowered, how do you make the most of that?
So there's lots of things. I mean, when we actually publish, like when you actually get to the point where because I've noticed a lot of the comments were like, you know, sometimes they need a couple semesters, sometimes they need longer, sometimes we need to add all the data together. Then you can actually, you know, probably conduct a actual statistical test where they can compare the differences, and maybe they compare maybe they use a statistical test that's appropriate, but they just don't have the power to be able to detect any differences. And then, it actually isn't a horrible thing to talk about, right? Because if they don't, but they can still, which is coming up in the later videos, they can still visualize their data, and they can start to see trends that are coming out even in their small replicates. And if they start to see trends and they just don't see it come out significant, which there's lots of issues with that, right? In a statistical test, then they'll know, like, what could I do better the next time, or what could the next person do? Like, if I had the time or whatever resources, I would add more samples, because then I would be able to detect a difference. But that's where you start. It's like a preliminary pilot study. I mean, there are so many medical, most drug trials start out with just a few people, right? Because they ask very, very specific, and they just want to see just the slightest bit of a movement. And so it's not definitive. Nothing's definitive until you've replicated it and reproduced it. But they're still providing valid research that is moving the science, you know, like moving the science. So it's all useful, even if it doesn't come out to be. And statistical significance isn't necessarily the gold standard either. Like we've talked about p-hacking, and I know Pam has been to my class where I made my students argue back and forth on whether or not the p-value was a valid measure, or was enough to fictitiously make her go get a patent, but it's because you can do p hacking and all these other crazy things statistically that isn't necessarily the end all be all. So I still feel like you have to support your results and your conclusions, just like I'll say in the video, with data visualization. So if you can, there's a lots of other things you can say about your small number of replicates.
I also want to add to that like when I published the paper with my students, he had many, many replicates, right? Because he worked over, he collected data for at least a year over 4 different seasons. But when I do a mini cure, I'm not going to try to publish the work after my first mini cure. Students will be adding to that so that we can compare seasons. So I could do the mini cure in, say, spring 2022, but I probably wouldn't publish that data until I have spring 2023. Now, what I do in that case is if students are interested in writing the paper, then they will keep in touch with me. We'll start working on methods and start putting the results in, and then they'll add to it so that I still keep in touch with the original students or whichever students want to participate in publishing a paper.
So there was a really interesting question about journals and page charges. And so the Journal of the Arizona Nevada Academy of Science will waive your page charges if you don't have a grant. You just have to let them know. I was I was president at one point and am a fellow. So that's a really nice positive place to publish. And they like observational studies. They are not so hung up on necessarily large sample size and replicates to begin with. You have an observational study and they'll, oh, that's interesting, and then you go out and you are able to identify some more funding to do the research. So we'll keep dropping some in the chat. And then there was another interesting question, which was a question for everyone. Who teaches student basic statistics using software, whether R or SAS or etc.? What are your challenges with that? So now we're back to the statistics again. And I tried to learn R and that didn't work very well.
Well, there are a number of ways. Base R or even RStudio has its own challenges. There are programs out there for biology instructors, biology based. It's called R Carpentry. They have a lot of workshops out there for R. Those are nice resources for, and they're mostly life science biology focused. However, just for students, I posted a new, it's actually for a textbook, and I'm actually using it for a different curriculum that I'm writing that's funded by the Bill and Melinda Gates Foundation. So it won't be branded with the book here in a little while. So, but these web apps that I just put are so user friendly. There are shiny apps. So our shiny apps. So these are the apps that will work on your phone. And actually, I just had a student, I know, I talked about this yesterday. I just actually had a student that for a publication used the app to create a graph because he wanted to do a stacked bar chart over a couple different groups and he wanted to manipulate the colors. And he was it was so nice. And he actually did it in because you can cut and paste the data from a Google sheet, you can upload a CSV, or you can actually just cut and paste, you can manipulate the labels, and it's all organized by app. So you can say, okay, well, explore quantitative data, explore categorical data, do a test to compare a proportion. It literally says these things and you click on an icon. So it makes it very user friendly for the students to use that app. And we're writing an entire intro stat curriculum based upon these apps because they are the absolute most user-friendly ones that I've seen out there, that are free, that aren't attached to a stat book per se. And then they also have, yes, R works on a Mac. So SAS doesn't work on a Mac. So that's why I have a PC, because I use SAS. SAS is way too difficult to use for an anybody student really. But R works on a Mac. R works on everything but a Chromebook if you because you have to download it. But you can use the cloud app for R on a Chromebook or anywhere, which is why most textbooks are using it, because it's the most it's the most universal, and it doesn't exclude anybody for any financial reasons. So because Chromebooks are kind of the way of, like, most students will have at least a Chromebook, but that's not very powerful in order to do any other computing on. So this app actually works on my phone, and I can download a graph from my phone. So it's pretty simple, and it makes it very, very easy to do, and very, and it provides graphs, it provides tests, p-values, the whole bit. So
we had a request in the chat there for links to those apps. Do you have to jailbreak your phone to get them?
I know I did put them in. I put it in the chat, but it was from me. So they're the Art of Stat web apps, and that's the home page, the landing page.
I've lost it. Could you put it in again? All right, so we now have places to publish with students is now also available on the workshop website. This is great. I love the way we're crowdsourcing this. I love it. Okay. All right, so, all right. And he just put an app we wrote with R Shiny so students can analyze multi-TOF data. Excellent. Okay, so more questions, comments? All right, so we did just put in the chat the Art of Stat web apps there, so that's great. Wait another 30 seconds or so to see if there are any other questions. Okay, all right, so you lost the link. I just put it in there again, artofstat.com/web-apps. Okay, so people are saying, thank you, that was really super helpful. Okay, most stats are written in Linux. To get that to work on a PC, I recommend Penguin as a Linux subsystem for Windows. Yeah, to me, a penguin is a flightless bird. But I can pipette the heck out of anything. All right, so now is a scheduled break. And so we will be coming back at 11:30. Is that right? Got too many screens open here. All right, so we are coming back at 11:30, and Kim Kobachek is going to be talking to us about her cure on the persistence of latent forensic evidence. Kim, do you want to give a little bit of an introduction so that when the videos start at 11:30, they are oriented?
Sure. Yeah. So the videos that that you're going to see kind of take you from the crime scene and into the lab the different techniques that not only did my students use, but that are commonly used in forensic science. So these are techniques that you can do with little to no equipment, and you'll see how they work both out in the field, or back in the lab. So I try to give you a little bit of background information about what stuff is going on in the crime scene as well as in the laboratory. The cure that I did was the persistence of latent evidence, which sounds so small, but actually, how long does invisible evidence last? Basically, that's what my students had to work with. Those are the types of questions they came up with. And so these techniques will help you uncover that invisible evidence. So I hope you enjoy the videos when you come back. Thanks.
Thanks, Kim. Okay, so we're going to go on break, and the videos will launch right in at 11:30 our time.
Hi, so we are back at the crime scene, and I want to introduce you to another common tool that is used both in the laboratory as well as at the crime scene that is used to identify and then help collect evidence. Now, we used a flashlight earlier to visualize this swipe of red-brown staining on the table. So we used it at an oblique angle to better visualize this potential blood stain that we are going to see. Now, the flashlight can, of course, also be used to look at your interest. Hey, look, there is a piece of trace evidence, this pink fiber. Let's take a look more, see what else we can find. Ooh, another piece of pink fiber. And then it looks like there's some staining on this chair. Well, the flashlight is great, but a flashlight only produces white light, which only has so much power, if you will. The use of an alternate light source, which you've probably seen on TV represented as a blue light or a black light or a UV laser, that's often used with these, really cool orange glasses, these barrier filters. This is the alternate light source. This is the useful tool that I'm talking about here. And this produces a different wavelength of light that will help the scientist better visualize evidence. Now, this particular alternate light source produces a blue light. Other alternate light sources can produce different wavelengths of light, but for now, we're just going to go ahead and use this blue light and demonstrate how much more detail you can see at a crime scene with just this different wavelength of light. So if you see here, this trace evidence, this pink fiber, this fluoresces, but this white-ish area here is of interest. It looks a lot larger and potentially looks like some type of pattern that we didn't see before with just the flashlight. And as we keep looking along the chair, we'll notice some other staining. Now, this is potentially blood. It looks kind of reddish brown, but the one thing I want to point out is that this blood or this dark stain does not fluoresce under an alternate light source. Rather, it quenches the fluorescence. So unlike what you see on TV when you hear someone say the blood fluoresced, this is not the case. But this is also something that may not be visible with just white light. Now, as I said, there are a lot of different types of alternate light sources and a lot of different uses for them, and that's something we will talk about when we go back to the lab. So let's head back there right now and talk some more about the alternate light source. Hi, we are back in our laboratory area, and in this segment, we're going to take a closer look at alternate light sources. And as you saw at the crime scene, an alternate light source can be used to visualize latent or nearly invisible evidence. And also what you saw at the crime scene, there can be different types of alternate light sources that can be used. These alternate light sources here are two examples of handheld devices that produce one wavelength of light. There are other devices that are larger and they're still portable, but a little bit more cumbersome to use that will generate other wavelengths of light. So blue wavelength is somewhere in between 350 nanometers plus. There are also other alternate light sources that will have filters that will generate in the infrared range and as low as in the 200 nanometer range, which can be useful for looking at latent prints that have been treated with a fluorescent dye. But what we're going to focus on here is the use of a handheld alternate light source that produces blue light. This is a very affordable tool, and we're going to first visually look at this lovely t-shirt that I have. that I purposely made so I'm not this much of a slob in real life. And as you can see, there's a lot of stuff on this. Okay, there's a lot of staining. There's this kind of orangey staining here. We have some ink, maybe some green ink here. There is some pink marker here. Have something red-brown, maybe it's blood, maybe it's chocolate, I don't know. And as we look further on down the shirt, we have some more of this orangey stuff here and some ink, and I don't know, you know, there's a lot of stuff on this shirt, but there's also more than meets the eye. So using an alternate light source can help you visualize materials that will fluoresce under that particular wavelength of light. And in this case, what we're looking for would be biological materials. There are certain biological materials, such as saliva and urine, which will fluoresce underneath of blue light. Now, as we talked about at the crime scene, TV shows that blood can also fluoresce. And as we saw at the crime scene also, that's not necessarily the case. Blood is actually absorbs or quenches that fluorescence, and that blood will look darker against the substrate that it is on. So let's take a look at this t-shirt using the alternate light source to see if there are any stains we're possibly missing. So I am going to be putting on my protective orange glasses. And the reason these are orange is because these orange glasses will filter out the blue light and allows me to visualize the fluorescence that is being produced by the interaction of this particular wavelength of light with these substances. So as we turn the light on, holy moly cow, you can see that this visible orangey stain fluoresces. Okay, well, what does that mean? Does that mean that this is some type of biological material? Well, not unless you count wing sauce oil as biological material. So just kind of showing you as an example that not everything that fluoresces is going to be of import or probative in your case or in research. So looking more at the item underneath of the fluorescent light, we see here there is a fluorescent stain. I'm going to take away the light here a little bit, and visually I'm not really seeing much of a stain. So this demonstrates, the light back on, that there is something here that could be of forensic interest. If we look here on this dark block, we also see that there is some type of off-white, crusty type of material. This could be indicative of a biological fluid, which will fluoresce under UV light. Now this stain here, I know it's blood because I put it there. Again, you're seeing it's not fluorescing. It's actually, you can see it, but it's not fluorescing. Okay. Now look at, oh wow, look at this here. We have this long stain here that is fluorescing. But again, I'm going to turn the light off, and looking at it visually, we're not really seeing that bright of a stain, if you will. We can see closer that something is there, but it's not as evident as it is underneath of the alternate light source. And again, flipping the shirt over, we're seeing some additional stains that could be of importance, but we're also seeing some visual stains, such as this orangey stain here, which is fluorescing as well. So again, just because it fluoresces doesn't mean that it's biological in origin, nor does it necessarily mean that it's of importance to a forensic case. What you'll also notice is that we can see some other stains here that fluoresce, that again, we take this off and they are not that visible, and actually they're almost whiter than the actual substrate themselves. Now this here is an example of bleach. So there are a lot of things that can fluoresce, even under the alternate light source. Bleach and cleaning supplies can fluoresce, as you can see, various inks, chicken wing sauce, in addition to biological fluids. So it can be important for you as the analyst to be aware of that. Now, if you're looking to see what types of biological fluids will fluoresce, we're going to take a common accessory that the trend started in 2020 and is continuing on into 2021. How long, I don't know. Saliva can be a very important piece of forensic evidence. And oftentimes saliva will not show up as fluorescent as, say, another type of biological fluid. But because these face masks are in such close proximity to the mouth, we can see that we're getting a nice fluorescence with this face mask. So this will enable the scientist to sample a more specific area to test for saliva than, say, cutting a larger area and maybe not getting that target area that you want to test. We take a look at these shoes, normal shoes, nothing up my sleeve. And if we look at these are also fluorescing. Again, perhaps this is not biological fluid. We can also see here that there could be some element of a pattern that could be present when we look at it with the alternate light source. Now, turning the light source off and looking at it with just our naked eye, we're not seeing that same level of detail. So as you can see, the alternate light source is a very useful tool. And it's useful not only for the detection of biological fluids in serological analysis, but it's also useful in different areas of the crime laboratory or different areas in which you might want to do an iCURE. Areas such as trace analysis, different types of fibers will fluoresce differently. In question document analysis, question document examiners will use light of different wavelengths to look at the different types of inks to determine if a document has been altered or not. It's also used in latent prints, as I mentioned, in addition to a fluorescent dye to help visualize latent prints even more. And lastly, it's also used in forensic photography with all different wavelengths of light. One example you see is here. This could be considered forensic photography. The videographer here is using the orange barrier filter to show you what the fluorescence looks like. That is done in the crime lab and at the crime scene as well. Infrared light, a different wavelength of light, can also be used in photography to demonstrate the presence or absence of gunshot residue or blood when it is against a darkly patterned type of substrate. And it can also be used to look at subcutaneous or under the skin bruising. So we've talked about this powerful little tool, which again
can be used either this small, or if you want to size up, you can do that as well. And we have one more test that we are going to talk about, which I've mentioned, and that will be latent prints. So let's go back to our crime scene and see what we can do with latent print collection. Hi everybody, thank you so much for joining me here today. My name is Kim Kobajek and I am a forensic scientist. Just to give you a little bit about my background, I have been in forensic science for well over 20 years, including being a practicing forensic scientist in a crime laboratory for both the city and the state. And I've also been a clinical associate professor here at Arizona State University at the West Campus, where we have a forensic science undergraduate degree. Now, forensic science, you may know that it's a very popular topic, and once upon a time, everybody didn't really know what the job of a forensic scientist was. But now with CSI, all of these different crime shows, now you have an interest in forensic science. You have, like I said, university degrees. You have concentrations that you can do, and even at the high school and junior high level, you have elective classes. So this means this is a science in which you can do research. But if you're not a forensic scientist, or if you're not familiar with it, how do you develop an iCURE to use with your students? That's why I'm here today, and I'm going to discuss with you not only the iCURE that I did with my students here at ASU West, but I'm going to introduce you to some common methods that are used within the crime laboratory, but are also easily used outside of the crime laboratory by instructors and students alike. So as they say, forensic science starts at the crime scene, so let's go to the crime scene. So this crime scene, we are going to be looking at different types of evidence. When you get to a crime scene, you really don't know what type of evidence you could be looking for. And oftentimes there is visible evidence as well as latent evidence. And in the iCURE that I did with my students here at Arizona State University, that was the focus of our research, was the detection of latent forensic evidence. Now that's a very broad topic, but if you think about it, latent or invisible can mean a lot of different things. It could mean blood that could have been washed away. It could mean latent fingerprints. It could mean trace DNA or trace evidence. So as I said, we're going to look through a few different procedures that, like I said, are usable in the crime lab as well as in the laboratory setting. So here in our crime scene, we're looking at some visible blood stains, and we can see fairly clearly on this bottle opener here the presence of red-brown staining. It also looks very shiny in appearance, kind of like a candy apple red on different areas of this item. But what might be less apparent is some red-brown staining that looks wiped on the table. So what I'm going to do here, this is just a regular flashlight. I am going to light this up at an oblique angle, or an angle that is perpendicular to the blood or possible blood, and you can see it just a little bit clearer on the table. And again, this is a common tool, the flashlight, that is used at the crime scene. So this here is also some possible blood on the knife. It has that similar red-brown characteristic. But one thing to point out is, in comparison to your bottle opener, the blood does look different. And that is something that is going to be observed both at the crime scene as well as in the laboratory. And we'll visit the laboratory later to kind of give you a better idea of how blood can look different on different surfaces, which could be an idea for a good research project.
So this looks like blood. How do I tell if it is blood? Well, one of the things that is commonly used at the crime scene, as well as in the crime laboratory, is an indicator test. These indicator tests for blood are presumptive only, meaning they don't identify blood and they don't identify human blood, but they are fairly specific to the protein heme, which is in blood, and gives the crime scene specialist or the forensic scientist a good idea that blood is probably there. Now, the test that is commonly used is the Kastle-Meyer test. And this is a 3-step test, and it's fairly simple. You take a drop of deionized water, and then you can gently swab a portion of the stain, which will then transfer a small amount of stain onto the swab. After you have a small amount of the stain transferred onto the sterile swab, you can start performing the Kastle-Meyer 3-step reagent test. And like I said, we'll visit the lab a little bit later and go into more detail about how this test works and what these reagents actually are. So step 1 is adding a drop of the Kastle-Meyer A reagent. And at this point, there should not be a color reaction seen. Then we will add the second reagent. A couple drops here. This will be a little bit harder to get out. And you want to make sure that you don't touch the tip of the swab to the reagent. Now at this point, we still see just that color of the stain onto the swab. And after we add this third reagent, reagent C, we should see a brilliant pink color indicating a positive reaction. So this is a positive reaction for the indication of blood. Again, it's not human specific. It is not specific to blood. There are things that can cross-react with this test, but the fact that this test may have some false positives is actually a good thing in terms of developing an iCURE. This is something that students can actually investigate further. So we saw, I'm going to perform one more Kastle-Meyer test. We saw that this was a pretty concentrated stain of possible blood. Now I'm going to perform that same test on that different appearance of blood here on the table. So again, I added a little bit of deionized water to the end of my sterile swab. I am going to transfer a little bit of that stain onto the swab, and then I am going to perform my 3-step Kastle-Meyer test with reagent A, reagent B, if you cooperate. You know, anything in science, it's not going to be perfect. That is all part of the experience. Okay, and then reagent C. And again, we have this bright pink color within 10 seconds, which is indicative of a positive, presumptive, or indicator test for blood. So we've done our tests, and now this evidence would be collected and sent back to the laboratory for further testing. Now, when we go back to the lab, we'll further discuss the Kastle-Meyer tests, some other indicator tests that could be used, as well as some common false positives that you want to be aware of when you're using this test. So let's go to the lab. Hi, and welcome back. We are in our designated lab space, and we're going to talk a little bit more in detail about the color indicator test that we use to detect the presence of blood. And if you recall, that test was called the Kasselmeyer test. Now that is just the name of the test, and actually the name of the reagent is a little difficult to spell. It's phenolphthalein, and if you need me to spell that for you, just send me an email. But phenolphthalein, or the Kasselmeyer test, is just one of a lot of different indicator tests that can be used either in the field or in the lab or in the teaching labs or your iCURE. Other tests include something called O-toluidine or tetramethylbenzidine. And all of these tests, including another one we're going to discuss later on, have the same chemical reaction basis. They're all oxidation reduction reactions. So for the color indicator test, your product of the oxidation reduction reaction is a change in color. Now this is a swab that we used in our earlier crime scene testing. And if you recall, it really wasn't that pink after we did the initial testing. And if you also recall, I said that a positive reaction within 10 seconds, this development of a bright pink color, is a positive. But as you can see, it's pink and even pinker. So doesn't that mean that this is even more blood? Not necessarily. If you go back to that chemical reaction, oxidation reduction reaction, you'll come on to the idea that if you leave these reagents out too long, they will oxidize. They will eventually turn pink, even in a negative test situation. So what I'm going to do to show you that is add our 3 reagents to a blank sterile swab, and at the end of this segment we'll see how pink this non-stained swab is. We'll put that here at the beginning. So just as a recap, here is a stain that was collected from our crime scene, red-brown stain. And I am going to add reagent A, which in this case is ethanol, reagent B, which is a reduced phenolphthalein reagent, and reagent C, which is 3% hydrogen peroxide. And here is our brilliant pink color, positive within 10 seconds, indicative of a positive Kasselmeyer test. Another thing to be aware of when you're developing your iCURE or testing evidence, blood can look very different on different surfaces that it's on. You typically think of blood as this bright red, shiny substance, but as you can see on this hat, this white hat, this blood here, which I've tested previously, shows more, it's a little bit more brown, a little darker, may not necessarily scream possible blood stain to you. Now this sock, which has a number of different stains on it, not only does it have some greenish stains, greenish brown, but also some red-brown staining here on the foot as well as at the ankle. Again, I know these are blood because I put the staining there, but this might be something that could be overlooked not only at a crime scene, but also kind of discounted in a research type of situation, only because it's not presenting itself as red-brown staining, I'm blood. And actually all of this this brown staining here, which looks like maybe somebody wore the sock on a dirty parking lot, this is actually wiped up blood. So you can see there's a lot of different treatments that blood could go through, and then you and your students could then in turn test to see what type of reaction you might get with the Kastle-Meyer test. Now this is a piece of carpet, and here is this red-brown stain, nice and visible, and then on down to the end of the carpet, the stain decreases in visibility, increases in dilution, decreases in visibility. Now, another pro to using the Kastle-Meyer test is its sensitivity. And there are stains here, as you can see, or maybe not. And a pro of the Kastle-Meyer test is it can actually test positive on a non-visible stain, which can be good and can be bad. In a research situation, that can be of benefit because you can further test how dilute can we get to get a reaction. But in a crime laboratory, it's not necessarily ideal because we won't know, can we get DNA analysis on this? Now these carpet squares here have examples of stains and substances that can look like blood. And so we're going to do a little testing here. So audience at home, kind of guess in your head which one of these you think might test for blood. I will give you the answers afterwards. So I'm going to wet a couple of swabs with deionized water, and I'm going to test our two stains here. I'm going to call this stain A. Make sure that I rub this nice hard to get a good color transfer. And here's the color transfer for stain A. Okay, I'm going to put that down. And then I'm going to swab stain B. Now stain B, probably doesn't look like blood, right? It looks a little orangey, it looks a little icky, crusty. And then here's the color transfer from stain B. Now looking back at stain A, this area here that's circled, that really doesn't look like that red-brown staining either. So you know what, we're gonna test stain B first, just to kind of get that out of the way. This kind of orangey color, probably not blood, but let's see. I'm gonna move some materials forward. I'm gonna add my reagent A, which is ethanol. Reagent B, the Kastle-Meyer reagent, reduced phenolphthalein. And then reagent C, which is my hydrogen peroxide. And I should see a beautiful pink color within 10 seconds if it is positive. I am not seeing that color. You don't know it, but I'm counting to 10 in my head. I am not seeing that color. So therefore, this is a negative test. It's a negative indication for blood. So in the crime lab, this sample would not go forward for further DNA testing. Now, much like we did with our blank stain, I'm going to put this negative result off to the corner. We're going to take a look at it after we complete our testing here to show you why you need to read these tests within 10 seconds. So just a recap of what we've done with this stain A right here. Not really looking like blood a lot. I'm not seeing a lot of red-brown. Honestly, it could be something like soda that's been spilled on the carpet. So I'm going to add my reagent A, ethanol, reagent B, which is the Kastle-Meyer reagent, and then reagent C, which is the hydrogen peroxide, Oh my goodness, look at this. Brilliant pink color, indicative of a positive reaction for Kassemeyer. On a stain that didn't really look like blood. So this kind of demonstration here shows you that the scope of what one can do for research, just involving blood or blood lookalikes, can encompass. There are some known false positives with the Kassemeyer test. Since the Kassemeyer test is based on an oxidation reduction reaction, there are substances that contain oxidizers, such as bleach, or other peroxidases, such as horseradish. Some other well-known false positives are some types of metals, okay? Now, when you think of bleach and horseradish, those don't look visibly like blood. But what if they were mixed in with some type of red-brown material? Could you mimic a positive reaction? So that's just one idea for an iCURE that can be done with something as simple as materials that you can get at the grocery store and the Kassemeyer test. So now let's look at our blank and negative samples. We'll show you here, especially our blank sample, there is some pink color change. And again, I just want to stress the importance of why reading this test within that 10 seconds is important, because these will continue to oxidize and just get pinker over time. Not only is the Kassemeyer a sensitive test, it's quick, it's easy, and you can actually perform it without blood if there's an issue with the purchase of biological materials. So just some food for thought for one idea for one of your iCUREs. So now let's go back to the crime scene and look at another method that can be used not only at the crime scene, but in the laboratory as well. Hi, we are back at our crime scene again, and I'm going to be introducing you to yet another technique that you can use in the development of your iCURE. And just to recap, we've already covered indicator tests for visible blood, as well as the use of an alternate light source to find latent or less than visible evidence. The next test that I want to talk to you about is also a blood indicator test, but it's used on less than visible or even invisible blood. Now, I'm sure that you've seen on TV or even on YouTube or some other type of media that when you spray something onto blood, it glows and it glows and it looks super cool. And that's what I'm going to show you here today. Now, this particular reagent is called Blue Star, and this is a proprietary brand, so it is not the same as luminol. But what's beneficial about Blue Star is that it comes in both training formulations as well as casework formulations. So we're using the training formulation here. And again, this is very easy to use, not only at the crime scene, but within the laboratory or teaching labs as well. Now, while there's many brands of both indicator tests as well as these types of fluorescent reagents, just know that Blue Star is but one of many different brands, and ASU doesn't suggest that you use any specific brand, nor do we endorse any specific brand. This just happens to be the one that I use in my laboratory for the iCURE. So as I said, Blue Star is used with less than visible or even invisible or latent blood. Using our flashlight again, let's start looking around the table to see if there's any staining we might want to test. Oh, look at this. All right, underneath I'm going to move this piece of evidence we tested before, and underneath here, there appears to be some type of pattern. And we didn't see that before, even when using our flashlight on the blood stain here. Now, looking in different areas of the table, oh my goodness, we can also see or illuminate some other possible patterns that our naked eye didn't quite catch. So what do we do now? Well, you can have someone shine a bunch of flashlights and take a picture, or you could use your blue star reagent to visualize this pattern. Now, the blue star reagent, as I said, is similar to luminol. It is an indicator test for blood, but instead of a color change, what you are getting with substances like luminol or like the blue star reagent is chemiluminescence, meaning that the chemical reaction produces light instead of a color reaction like you get with Kassemeyer or those other tests that we discussed. So let's try this potential area here and see what we can find. So what I'm going to do, I've already made the blue star reagent according to the instructions. And then I am going to spray this reagent onto the potential pattern, potential blood stain. As you can see, there is the creation of this luminescence, this light blue color, indicating the presence of blood. Now here, on this side of the table, we can see some small, visible red-brown stains, but that might not be the only staining that is present. So I'm going to use the blue star reagent to see if I can visualize more of this possible pattern. Again, I'm going to spray it gently onto the possible pattern in the entire area. And indeed, there is more than just the visible blood stains. So you can see here. You may be wondering, with all of the spraying of this reagent onto these blood stains that were invisible, how do you collect the blood stain? Or does this reagent impact any type of downstream analysis on the blood stain? Let's go back to the lab and we'll discuss that further. Hi folks, we are back in our designated lab area and we are going to be focusing on the second blood indicator test that we talked about back at the crime scene. Now, if you recall, this second blood indicator test that we discussed was called the blue star reagent, and it was similar to a luminol reagent or other reagents that will cause chemiluminescence or the production of light when the reagent reacts with blood. Again, it's not specific to human blood, and it can cross-react with things. But one of the reasons why I use Blue Star training materials in an iCURE is because for downstream analyses, let's say I have another student who wants to further test Blue Star blood samples. They can do that because it has been demonstrated that Blue Star, while it does dilute the sample, it doesn't necessarily affect the actual components of the blood that could affect downstream processing. So again, Blue Star is but one type of chemiluminescent reagent that can be used. Luminol is another one. Fluorescein is another one as well, but I'm just happen to choose the Blue Star reagent. This does work on the same chemical premise as the other color indicator tests that we discussed earlier, such as Kassemeyer and tetramethylbenzidine. It's an oxidation reduction reaction, but instead of a color change, light is produced. So it's called a chemiluminescent result. And again, the pro, or the usefulness of the reagent, is that it can be used to visualize less than visible or non-visible blood on different surfaces. So as you see here, we have some different substrates. We have some carpet with visible staining that starts to give way to non-visible staining. And we have some vinyl tile, which also appears to have some staining on it in various amounts. We also have this large carpet square, which really doesn't appear to have anything on it at all. Now, this is where your chemiluminescent reagent can be useful at a crime scene and can also produce some pretty imaginative hypotheses within an iCURE project. So let's go ahead and do a demo of the blue star on the visible stains, just to kind of show you what type of luminescence or chemiluminescence to expect. So I am going to just spray this lightly on the visible stain. And as you can see, in these first two stains, another useful trait of the blue star reagent is that this luminescence can be visualized in less than dark conditions. There are some other reagents that require complete darkness to view the chemiluminescence. And also you can see the chemiluminescence lasts a little bit longer than maybe you've read luminol lasts or fluorescein lasts. So now let's keep spraying and see if we can visualize these other less than visible stains. Which we can here. Now maybe you're thinking, it's carpet, it's absorbent, of course the luminescence is going to stay. Again, that's a valid hypothesis. But let's look at this vinyl tile and see what the luminescence ends up looking like after we spray it on both the visible and less than visible stains. And as you can see, the luminescence looks a little bit different than it does on the carpet, and that's because we have a different substrate. We have a non-porous substrate versus a porous substrate. So here we see the luminescence actually lasting a little bit longer simply because it's not being absorbed into that porous substrate. Last, we're going to take a look at this large carpet square, which has no apparent staining on it at all. We might see some small stains here and here, but looking closely at this carpet square, even with an alternate light source or even just a white light and a flashlight, it's going to be very difficult to see because this is a patterned type of substrate. But let's use the blue star and see if there's anything we can discover on this carpet square. Hopefully, you're able to visualize that there is indeed some type of blood stain pattern or blood stain on here that can be detected. It may not be in your face obvious, but again, in a darker room, this might be a little bit more useful. So even with not complete darkness, we have on just an emergency light in here now, you can still see the fluorescence, and it does last quite a long time. This was sprayed a few minutes ago, and the lights are dimmed, and I'm creating shadows with my hands, and you can still see the fluorescence. So this is Blue Star. Again, this is one type of reagent that can be used to detect latent or nearly latent blood. It does have the ability to be used for further downstream testing, meaning that these samples here could be swabbed and collected to see what type of DNA profile could be perhaps generated from them. And then also the chemiluminescence can also attempted to be measured to see if dilution and brightness have any type of correlation to one another. So those are just a few ideas for an iCURE involving chemiluminescent reagent. So now let's go back to the crime scene and look at another common forensic technique in latent print development. Oh, that's a good one. Oh, hi. You caught me back at the crime scene using my alternate light source and looking at one of our pieces of evidence for the presence of latent prints. And indeed, I found some really good latent prints on this glass. So the next step is to do some black powder, right? Yes and no. Typically, the black powder that you see on TV, in this very fine black powder with one of these little delicate brushes, can be done at the crime scene on non-porous items. However, items such as this glass, which can be transported to the lab in a more controlled environment, are typically collected and then processed at the laboratory. Now, that's not to say that you can't do some powdering at the crime scene, especially on pieces of items that cannot be collected or may be in danger of losing evidence if they're being transported, say a vehicle, for instance. Okay, so to get our best evidence, let's take this back to the lab and powder it there. Hi, welcome back to our laboratory space. The last technique that I want to talk to you about for the collection of a particular type of latent evidence is fingerprint powdering. Now, as I discussed at the crime scene, this type of evidence here would typically be collected from the crime scene and then taken back to the lab for processing. Fingerprint powdering can be done at the crime scene, but typically it's only done if the item can't be moved. Something like a large table that we had in our crime scene. Another example might be an Arcadia door, a large glass window. So those are the types of items that you would expect to see powdered at a crime scene. And other items will be collected and then taken back to the crime lab. Now, I also mentioned that if something was in danger of perhaps losing that piece of evidence, maybe a vehicle, if it has to get transported back to the crime lab to be processed there, fingerprint powdering and some collection can be done on site as well as at the crime lab. Now, there are a lot of different methods of latent print development, but I'm going to be demoing the powdering method for you today, and we'll talk about a few more after this. Now, just as with the other products I've used, you see some brands here. This is not an endorsement. These just happen to be the particular brands that we're using in the laboratory. And black powder, I better not spill this because I will warn you, this is messy. This is a very fine powder, and if you get it on your face, it's really hard to get off. It's not dangerous, it's just kind of a nuisance. So I'm going to be very careful. And a little goes a long way. So when you powder, you're going to use this very fine brush, and you don't have to paint it on. You don't have to slap it on like you would paint.
It's very gentle. So if you think about applying powder to the face, or maybe gently petting a dog's head or a cat, you're going to be that gentle with the brush, with the powder on it, on the piece of evidence. So I'm going to dip the powder a little bit, and you can see with just that small dip how much powder I actually got. So I'm going to make sure that I get some of this powder off because I don't need that much. Then I am just going to gently swirl the powder across the glass. And in the crime scene, we saw, or at least I saw, some good latent prints for collection. Now, as I was saying, a little goes a long way. And if I need to apply more powder, I can. And I might do that just a little bit. This small area here, just so you all can see this at home. And perhaps we can see it here. These nice two prints that can then be tape lifted and placed onto a fingerprint card for later comparison. Now, I'm going to put the lid on my powder because I do not want to accidentally spill this. And I am going to use a hinge lifter or a tape lifter to attempt to lift these prints off the surface of the glass. Now, these are a particular type of tape that can be used to lift latent prints. However, in my iCURE, you can use good packaging tape. So this is taken the sticky side in front of my face. Now the sticky side will now be applied to the glass gently, and you just want to use some caution with it because you don't want to lift it, replace it, lift it, replace it. And even this probably wasn't the greatest job, but we'll see. Smooth it down to make sure that you've collected all of that detail that hopefully you will be able to see on the card, and then you will lift it and then place the tape onto the fingerprint card, and then this portion can then be discarded. So I didn't do a great job. That's okay. It's trial and error. This is why packaging tape can be a good alternative to the more expensive hinge lifters, because obviously it's a lot cheaper. But you can see here I did lift at least some pretty good ridge detail in black powder from those two developed prints that we saw on the wine glass. So then these can be analyzed and compared if there is any other known fingerprint comparison to be used. Here are some other examples of lifted prints, either through my previous teaching or others' teaching, and you can see they vary in quality. That's going to happen. That is typical. And it's typical because of experience, but it's also typical because when you're powdering a latent print, you really don't know how much ridge detail is actually left. So what you're seeing here are actually amazing examples of lifted latent prints. And you might think, oh, these are kind of crummy. Well, you should wait until you see a crime scene latent print. You're not necessarily going to get this good of quality detail. Okay, so I've talked about powdering, and I've mentioned black powder.
if you have a different type of surface, say a dark surface, there's also fluorescent powders that can be purchased, as well as white powders and gray powders. So you're not limited to just black powder. It's, again, going to depend upon your surface. Let's say that powdering or fingerprint powdering isn't really something that you want to use as a standalone. There's also something you've probably heard of called superglue fuming, and actually the more appropriate term is cyanoacrylate fuming. And yes, the reason why it's cyanoacrylate is because that is a component of superglue. And the cyanoacrylate in a closed chamber that's typically heated will adhere to the oils left by the latent fingerprint. And of course, in a crime laboratory, you have purchased cyanoacrylate, but instructors and science lovers at home can also use this technique and develop a latent print at home. And what we found here is that if you use a little bit of superglue that has been placed in some tin foil at the bottom of a large jar, say a mason jar, or even a large pickle jar, so eat all those pickles that you bought from Costco, clean that pickle jar out, and you can actually put things like spoons, our cutlery that we had at our crime scene, and put a few drops of that super glue onto that tin foil that's been placed at the bottom of that pickle jar. Seal the jar, and it takes longer, but you can actually see a print develop using your homemade super glue fuming chamber. So that's just another alternative that you could use for an iCURE that's very inexpensive and less equipment oriented. And lastly, I want to mention that these two methods, the powdering and the superglue or cyanoacrylate fuming, are for non-porous substances. So these are not going to be used on things like paper or magazines. So if someone had a threatening letter, you're not going to powder it, you're not going to superglue fume it. There are processes and reagents that are used for non-porous substances, and those include reagents such as ninhydrin, as well as iodine fuming. And I know iodine really isn't the greatest chemical to use, but it's also fairly inexpensive if you get it from a chemical warehouse, and as long as you handle it safely and according to protocol, you should be okay with that. The iodine fuming is typically an older method, but it can be used, and it does produce very nice fingerprints on porous material. So I've given you a lot of information, both at the crime scene and in the laboratory setting. I hope this has sparked some ideas. I hope this has given you some useful information that you can take back to your institution or to your schools, to your students, to help you develop some really cool research projects in and around forensic science. If you have any questions, I know that we've all provided our contact information within this workshop, and I do hope you will contact me with any questions. This has been a lot of fun, and I hope you've enjoyed yourselves too. So thank you again. I'm going to go back and finish processing the crime scene, and we'll see you later. Bye.
All right, I was just remembering one time when we apparently had processed one of the teaching labs as a crime scene, and whoever processed it did a great job. They pretty much black powdered every surface known to humankind, and then they forgot to clean it. So you put your book down and then it's there, you touch your face, and all of a sudden there you are on Mary Poppins, like a chimney sweep. So that black powder gets everywhere. And I guess we're having a little bit of a discussion about whether black powder or glitter would be worse to get everywhere. And I'm going to have to say black powder, because when we did it, it got everywhere, and it was really hard to clean off. But perhaps the others, want to counter me, and then we'll get into the questions.
Yeah, glitter, you can at least tape lift, or you can vacuum it up. Black powder, you can't do that. I mean, you have to, it was awful. It was just awful. I remember that it was in one of the bathrooms too. And one of the bathrooms had this tile wall with white grout. And I don't know why the students, well, that's neither here nor there, but yeah, black powder, worse, definitely.
Oh my gosh. Yeah. So I was a victim of the black powder massacre, the explosion.
Mm-hmm.
So yeah, I would really caution people, if they're going to use black powder, it really is very difficult to get off of things, and it gets everywhere. And I had a big smudge, and yeah, I'm sure my students were laughing at me. All right, so it's time for question and answer. We have lots of people saying it was great videos, and then we had someone asking if maybe later on could be also opportunity to meet. Absolutely. I think our contact information is on the website, so we're happy to keep in contact. So questions about these forensic science videos or anything else?
Oh, yeah, thank you. The techniques were really fun to do. I wanted to do this to give folks who haven't done any forensic science stuff kind of an idea, really, of kind of how straightforward it is. So while we're waiting for questions, if you have questions or have comments, that's great. But it was kind of the idea. Oh, hi, yeah, hi, Dr. Sweat.
Kim, could you describe for everyone some examples of student questions and student projects that you had in your forensics class, and some of the diversity of ideas the students came up with?
Yes, I had. Now this was a great class. Like I said, it was the first summer cure, so it was 8 weeks long. So students knew they had to come to school in Arizona in the summer. Now, these were all local students, but, going out of your house in the summer in Arizona is a challenge enough. And I had students who voluntarily set up 2 experiments outside that they had to check daily. So let's see, I had 1, 2, 3, 4 groups. Some groups had one person in it. And I am reminded that there was a hair question, and I didn't quite see what the question was. So I can go through the questions my students had, and then I can address the kind of the trace evidence hair question. One student wanted to know about persistence of latent prints, development of latent prints on hair. Identification of hair samples from the crime scene. Okay, let me. Obviously, I can't walk and chew gum at the same time. Trace evidence here, so I'll address this one here. Trace evidence is a really good example of something you can do for a cure. It's less equipment intensive, and really there's a lot of different hairs that even just a compound light microscope you'd be able to look at. You don't need something fancy like an SEM scanning electron microscope. Even something like a stereo zoom microscope would work too. So yeah, I could definitely see where the identification of hairs or different types of hairs, different types of trace evidence that have been collected from a scene, or the persistence of such trace evidence collected from a scene or a particular scene over time. You know that you've planted a certain amount of hairs, and how long do they stay over time? That's a great idea. That's a great question. Now, with forensic science, hair has a little bit of a different history, and mostly, hair comparisons aren't really done now. But for a good research project to get them started on a question, I think that's a great one to have. Another example of a question one of my students had was the persistence of saliva on bodies. So this is where I found out, okay, students want to do some experiments on dead bodies. Why? Well, believe it or not, there is actually latent evidence left on victims who have been, of course, left if they're dumped in the desert or left at a crime scene over time. So a group of students wanted to know saliva can be present in bite marks and in other types of situations on a body. So that's where I had to get the rats or the mice to determine how long this saliva could be detected on these bodies that were left outside in the Arizona heat for a certain amount of time. And one of our nuisance variables that we didn't take into account, even though they had a cage, they built a great cage out of chicken wire, they hammered it down. We have some really smart coyotes or really smart feral cats around here, and they got in. And they had to deal with it. Another question dealt with the persistence of bloodstain patterns in the heat. And then, another student group did the persistence of blood after it's been cleaned and painted over. Again, trying to mimic, okay, well, what would someone at a crime scene do? Okay, well, blood gets on the wall. Okay, well, I'm going to clean it up on the wall. But I'm going to paint over it too, because you may not have seen it very well in the videos, but the luminol or the blue star would be able to detect the cleaning. And they did some really good experiments on different cleaning materials and different types of paints, and how those could or couldn't be detected with blue star. And 2 of those papers or abstracts were actually accepted for poster presentations at the American Academy of Forensic Sciences, which is the international forensic science professional organization. And so they were able to go to that conference and present. And I was very, very proud of them for that. So again, as undergraduate students, they attended a professional organization, met a lot of different people, and they have published proceedings. So I could just talk about that forever. Let's see if there's any other questions here.
Didn't you have a couple of other projects? Didn't you have a fingerprint project?
I did, and the fingerprint project, unfortunately, that was one of those situations where it was kind of an example of maybe what not to do for the future. The student really wanted to do this. He really, really, really wanted to do this, and even the slightly being persuasive of, hey, why don't you try doing this? Why don't you try doing that? And after the first bits of information or non-information, if you will, came in, wasn't very successful. But I told him, these are still results. You are still getting results. And so this is something that, you're still going to present. So while it wasn't a successful experiment per se, I still think it was successful in that it showed you how to improve for the future and what could have made the question, I guess, better to look at. And he understood, he got it after a bit. So that made me happy.
So we did have another question in the chat about how much experience do you think some of these projects would need? So could we do a project like this without too much experience?
Yes, and I wholeheartedly say yes, because 2 out of the 8 or so students that I had were biology majors. Or biology and 1 environmental science. They had no interest in forensic science whatsoever, or they did, but they didn't have any previous experience in it. And then the students that I had were kind of in the middle of their forensic science degree. So they hadn't gone off into these 400, 500 level classes. They had still taken a couple 200, 300 level classes. So you don't necessarily have to have years and years of experience, or even 3 years of schooling to do this. The one thing I would say is, when you are developing something like this, look at what's been done. These, I have to say, the forensic science papers are really good about spelling out methods, materials. But then also, the biggest thing with forensic science is not necessarily materials and methods, but also the thought process that you have to be able to support this in court later on. So that's something else to get the students to think about, that documentation, being transparent in what's going on, that's also part of forensic science, which can also be part of your forensic science iCURE. So yes, there's a lot of companies, and I believe Jennifer Hackney Price put on the website the list of companies that have different kits, different sets that you can purchase, whether you're involved in crime scene forensic science or not. As educators, you're able to purchase these materials. And they come with instructions, they come with protocols. And if you're ever at a loss for where do I find a protocol, you can always contact me, really. I hope that answered the question.
I tried to roll some fingerprints.
Rolling fingerprints are so hard. It's almost better if you roll your own, because you are trying to control someone else's finger. And what do you want to do when someone has a hold of your hand? You're pulling away. So it's almost easier just to do it yourself, or just hit it, just do your own print. Rolling prints is a nightmare, and it's a nightmare even for fingerprint technicians to do it. So that makes anybody feel any better. It's kind of a challenge.
I was trying to roll prints with some elementary students. We were doing a little bit of, forensic science, activities, which were more of the, which was more of the inquiry. And I put into the chat an inquiry book that I really like, that is for K through 12. Remember, I shared those books with you. And they have a lot of forensic science for kids and so forth, and they're more inquiry, but they have some pretty simple ideas that then you could expand on much more.
Yeah, there are some really good books out there. When I first started at ASU, and let me mention this really quick. I mentioned that there were some websites up on the website, some vendors up on the website. I didn't get those in time, so those can be sent out to everybody. who's interested. They're just a list of links for vendors and for different ideas of where you can go for these forensic science specific products. And as Pam was saying, the books that she gave me and then prompted me to search out, there are some really good books out there. Even the ones, it's homeschooling science experiments, and it's great because they have a list of all these different supplies, where you can get them, very inexpensive. So when we're talking being on a teacher's budget, this book, I'll do a little bit of research on what the name is, and then I'll be able to put that up in the chat for everybody, because it's a really good book. And these are good experiments they seem pretty simplistic, but they can be expanded upon.
Great. Yeah, I found that when I was doing certain things with outreach with some elementary school students, some of the things were simple, but you needed practice, how to roll a fingerprint, how to look at a fingerprint, what is a ridge and all of that. And so, a little bit more practice.
Yeah.
All right. So we put in the chat links to the website with some of the projects, and then those 2 posters that you were talking about. So the students have presentations and they are in a proceedings. So more questions about forensic science?
Let's see, in the 8-week course, do you ask your students to be in the lab each week? Yeah, they are in the lab each week. For an 8-week summer course, the students have to attend, 2, 4, 6. They have to attend, I think, at least 6 to 8 hours of class a week. So instead of having them be there 1 hour for a lecture and then 5, 6, 7, 8, however many hours for a lab, we kind of the way that I did it was have a little bit of talking in the beginning, have maybe 45 minutes of lecture or so, and then take a break. Nope, no break. Great, come back and let's do lab stuff. And then we dove into the cure, and we used those worksheets that were developed, started talking about hypotheses and experiment development. That was the time when the students would also work on finding their source literature. So they were, like I said, I was very impressed with them. They were there every day, 2.5, 3 hours, 3 days a week in the summer, uphill both ways, So yes, they were in the lab.
Okay, excellent. So we have the link up now in the chat, link for the forensic vendors. And so hopefully that will help you think about this. And don't forget to fill out your survey. If you're a non-ASU participant and you fill out all your surveys, then we have generous support from the National Science Foundation to help you purchase a few supplies for your cure. All right, so more questions?
If there aren't, or if there's any questions that come up at all during break or whenever, like I said, please don't hesitate to reach out.
All right, excellent. So we are going to break for lunch until 2 o'clock. But before we do that, we want to tell you a little bit about the final videos that we are going to be showing. So we will show a few more videos and then have another question and answer session and then a conclusion. So that will begin at 2 o'clock Arizona time, and Jennifer Broach will talk a little bit about those videos.
Yeah, so the upcoming videos are just a continuation of what you've already seen. So there's a process that the students could go through from research question to analysis. So I kind of just go into basic experimental design. I mean, we don't go through everything. And then data visualization activities that you can introduce to your students, all of which are supported by the handouts. And then we have the final one was why stats. So it's kind of an interesting kind of spin on why you would even need to use statistics in their analysis, because not often students really, I think of statistical tests, but they don't always think that they need that to show any results or evidence. So it's just a little bit more about how you can support your students through the data analysis portion. And so we wanted to make sure that we had the time of the video correct. So I'm going to kick it back to Pam for a minute.
Okay, so the time of the video, 2 p.m. for the next videos starting. That's what I have on Run of Show, is that we have the basic experimental design starting at 2 p.m. Arizona time. On the website, it says 1:45. We are going to fix that to 2 p.m. So we will get back here at 2 p.m. All right, and we did want to one more thing. We just got the Citizen Science Bee Monitoring Guide with an identification section and with a web address, and so those will be in the Google the Google folder where everything else is. So we'll be back here at 2 o'clock Arizona time. 2
and 2.
So this video is entitled Basic Experimental Design. So we're going to move past the pre-planning process into the actual design of the experiment. So again, I'm going to reiterate why we're even designing the experiment is so the student researchers can make valid and objective conclusions. So we're going to again ask them to ask the right questions, which we did in the last video. And now we're going to make sure they collect the right data. So phase one is the pre-planning process, and that is done before designing your experiment. So they've already developed a testable research question. They've already identified their independent and dependent variables, and they've already identified the other variables that might impact that dependent variable. So they have already identified all of those key aspects that'll help them design their experiment. So in the actual experimental design, they're going to understand how basic design principles work. They're going to be able to identify key aspects in the planning process that transfer and dictate the design. And they're going to ultimately sketch out their experimental design by the end of these activities. So they're going to translate the things that they identified in the pre-planning process into the actual design, and they'll learn how to control for those other factors. They'll learn how all of those pieces start to come together in a design of experiments. I wanted to provide a quote by Doug Montgomery, a very famous design of experiments expert. All experiments are designed experiments. Some of them are designed well, and some of them are designed really badly. The badly designed ones often tell you nothing. So we want to avoid students falling into that trap. We want to avoid getting them to a situation where they can't make those conclusions that they want to make about their research question. So it is important to design their experiment well. So we've moved beyond the factor identification and the hypothesis identification to experimental design. So we're in the second aspect of our from planning to conclusion process. The key experimental design aspects that we're going to talk about are randomization, replication, and comparison. So the first one is randomization. We randomize to prevent selection bias. So selection bias means that one group is different than the other group by some way I've selected the two groups. So if, let's say I'm testing an additive versus a non-additive. I need to make sure that my experimental units going into each group, either they're getting the experiment or they're getting no experiment, they're the control group. I need to make sure they're equal in all other aspects. They need to be the same in all other aspects. And how we do that, typically with human trials, things like that, with people, with animals, with things that we have natural variation, we want to randomize them to two groups. So we will randomize your experimental units into the control group or the experimental group. They'll either get the treatment or they won't. But the key here is that if you randomize into one group or the other, then we mitigate all of the other bias that could come from the factors that we can't control. So typically these are for human factors, animal factors, things like that, that have natural variation. Variations in weights, variations in heights, variations in other things that may impact your dependent variable that you just don't know about. That's why we randomize into two groups. There are situations where you don't actually need to randomize. Sometimes in an experiment, we already know that all of our things are the same. For example, we order cells, and the cells are the same no matter if we put them in the control group or we put them in the experimental group. So regardless, they're going to be the same. So we don't need to randomize into those two different groups. So really the randomization comes in when you're talking about people, you're talking about animals, things that other factors can impact. So the second experimental design basic that I want to emphasize is replication. So what we're calling replicates, the number of experimental units that you have. So more replicates provide better information. You can't just do the experiment on one person, or you can, but it just doesn't provide as much information. So it's better to include more people, more cells, more whatever you're using as your observational unit. You want to include more of them. You want to replicate your experiment to see if you get the same results, or maybe you're getting different results. So obviously, the higher the number, the better. But this is often constricted by budget and time constraints. So even though there is a statistical way using a power analysis to actually figure out how many people or samples you'll actually need, that's usually out of the scope of an iCURE. We'll just usually stick to time and budget constraints, and you're just going to get as many as you possibly can within those constraints. Finally, we're going to compare. So the last aspect of experimental design would be to compare the two groups. We want to give them different treatments, and then we want to compare. So we want to randomize, replicate, and compare. So in the activities, we provide students with some simple experimental designs that likely their experiment is going to take. So hopefully they'll be able to identify the design that's best suited for their variables. As a reminder, students already have identified their variables of interest and their response variable. So they have already identified all the key aspects to this experimental design. So you start out with the same units, then you randomize into two different groups. One's maybe manipulated, one's not manipulated. And that's manipulated by your factor of interest. Okay, so that grouping in this center is your factor of interest. Then you conduct your experiment. And as a reminder, you've already identified these variables. As a reminder, you're going to need to measure your dependent variable, and you're going to measure the other variables that you decided that you've identified in the beginning in the planning process that could potentially affect your dependent variable. And then ultimately, you're going to compare. So another design that's fairly typical in an iCURE is the pre-post design, where you start out with all of the same units, you apply the treatment, and then you compare them. You collect the information and then you compare them before to after. So for example, we give the students an example in an activity. So we ask them to identify all these different factors and insert them into their experimental design. But part of that idea generation is to show them a simple experimental design so they can start to see how their experiment will compare or contrast to a simple experimental design. So, for example, this research question is, does a new medication lower the low-density lipoproteins or the LDL levels of cholesterol more than the current medication, controlling for diet and weight? So this is a nice example where they've already identified the dependent variable, the cholesterol level. That's that objective measure of whether or not the drug worked or not. Our factor of interest would be the drug, and we're going to split that factor of interest into a control group, the old drug versus the new drug. And then the last statement states controlling for diet and weight. Those are those other factors that the student identified as potentially affecting your dependent variable that they're going to measure. So they start out with their experimental units. They randomize into two different groups. One group gets the old drug, one group gets the new drug. You'll notice that I added the sample size, but the number of students in each group. So the students start to think about how many replicates they're going to need for that part of the experiment. Then they're going to conduct their experiment. So then they're reminded in their conducting the experiment to collect the response variable and to collect all of those other variables of interest, i.e. here, the diet and weight variables. And then ultimately, they're going to compare the cholesterol levels between the old drug and the new drug, and that comparison will help lead to their conclusions. Data visualization and analysis. So you've already done your pre-planning and the experimental design, and students are either collecting their data currently or they've just finished their data collection before they move on to data visualization. So again, we're designing the experiment so the students can make valid and objective conclusions at the very end. So we've already asked the right questions. We're already collecting the right data or on the path to collecting the right data. And now we need to analyze the data correctly in order to make those valid and objective conclusions. So we want to be able to use data to support our conclusions. So we're going to build upon all of the phases that we've already done and utilize data visualizations and statistical methods to support the conclusion the student wants to make. So in this phase, the students will identify what's good and bad about graphics and data visualizations. They'll learn how to appropriately visualize data through a number of activities. They'll also recognize the need for statistics in this role of statistics activity, which is actually the topic of the next video. And they'll be able to identify and select the appropriate statistical test and ultimately evaluate their conclusion. So we've already done the planning process. So we've have all of these materials leading up to the analysis and conclusion. So all of the activities that are provided for you on the workshop website all go through and support the analysis and conclusion. So data visualization is your very first step in data analysis. It should support the conclusion of your research question. That's why data visualization is so important, is that visual to support your conclusions. So the next activities will help build a student's intuition about what makes a good and a bad visualization. What are the key aspects of a visualization that they should look for in making their own? Like labeling appropriately, the use of color, what types of graphics are best for certain types of data. We want them to explore a bunch of different options so they know what's out there and we're not limited to pie charts and bar graphs. We'll also help them develop appropriate graphics and learn to see what they would be best for their data. So for example, one of the data visualization activities asks the students to rank from novice to emerging to exemplar of all these different aspects of a graph that make it a good data visualization. Because the better foundation of good versus bad data visualization will really help the students create their own. So the next activity that really helps support their thinking in their own data now, before they're looking at other people's graphics, well-designed commercial graphics, commercial plots. There's lots of different options out there to look at different plots and provide different opportunities to have your students look at different contexts. But the final one brings them back to their data. So we're going to ask them to draw their own hypothesis. So what does that mean? That means you want them to think about the data that they're collecting and what it's going to look like. So this is while they're doing their experiment. There's usually a lull in your class where they're collecting their data and they haven't really gotten to the point where they're going to actually analyze the data because the data collection isn't over. But they've started collecting their data, so they know the types of data that are coming in. And so you want to ask them to draw their hypothesis. What is your data going to look like if you can show differences between the group? What is your data going to look like if you cannot show differences between the groups? So you want them to anticipate the data that they're going to collect, and you want them to anticipate the types of graphs that might be appropriate. Because if you're looking at whether something is present or something is not present, that graph is going to look very, very different than if you're comparing two means that are numbers, right, that are numeric in nature. So we want to make sure that they're choosing the appropriate plot for the type of data. So this draw your hypothesis activity allows them to internalize and use their own data in creating these fake graphs. They're not going to actually plot the graphs for real. They're going to just sketch them. What are they going to look like in theory? So there are a lot of tools for data visualization out there, and I'm going to present just one of them. One of them is from an Art of Stat website. It's actually through a textbook, but they're free apps that are run on R Shiny. So R is very intimidating to create any graphics, as a science student, or as really, I mean, even as a statistics student, they're very intimidating, and programming in R or some other software is oftentimes very daunting. So there are a lot of apps out there that create visuals very easily for the students. For example, this is just one of many tools that we provide you as a resource. So this one is about exploratory data analysis that will allow them to quickly visualize their data, whether their data is categorical, whether their data is quantitative, or whether their data is over time. They can select the type of data that they're looking at, and it will help you out with a number of different graphs. Like, for example, the Explore quantitative data will do side-by-side histograms. They'll do side-by-side box plots. They'll do side-by-side density plots. There's lots of different choices within the app for your students to use. They have built-in examples within the app, and also there's also the way to cut and paste your students' data into the app very readily. It prompts you to enter data, and all you have to do is cut and paste, and it's there. And the students can create plots in R, but in a very user-friendly way. So the next video is the role of statistics activity. This is after you've done your pre-planning, you've planned your experiment, and now we want to move beyond data visualization to statistical analyses. And statistical analyses, whichever one's appropriate, is well beyond the scope of this video. But what I really want to do is help you motivate the need for statistics in supporting your conclusions with your students. So we want the students to be able to support their conclusions through the proper use of statistical methodologies. So that's what we want them to know. We want them to recognize the need for statistics. And why can't I just stop with this graph? I can show the differences with this graph. I've done this great job with my data visualization. Why do I need to go a step further? And we want them to recognize that need for statistics and appropriate statistical test that moves them beyond their data visualization. So again, we're still in this last aspect of the analysis and conclusion portion of our iCURE. So the students have already conducted their experiments, and they're just putting their final touches on their posters, and they're making their conclusions. So we want to emphasize that they have to justify their conclusions of their research beyond visualizations. It's not enough to show that the bars aren't the same height. It's not enough to show that these two numbers are different. We need to help them understand what role statistics have to provide evidence beyond chance. So this activity is kind of fun. So suppose you're trying to prove if somebody has extrasensory perception, so ESP, so their sixth sense, you say, so their ability to read your mind, right? So early on, this psychologist developed these cards that had different shapes. The colors are relevant here, but they had different shapes. And he would present a subject that claimed that they had ESP with these cards. And they were obviously face down, and they were supposed to tell what shape was on the card just by seeing it face down. They can't see through the card. They need to be able to have that sixth sense or that extrasensory perception in order to guess the right shape. So suppose you're doing this with your experiment, and you want to prove if somebody has ESP. So you give them the card, you put the card down, you pick one of the cards out of the stack, you put the card down, and your test subject can either get it right or they'll get it wrong, right? So there's two options here. And so suppose you give the card to this person that claims they have ESP, you give them the card and they guess correctly. What if they guess correctly? Can you conclude that they have ESP? Would you conclude that if they have ESP? So these are the questions you would ask your students. So you're going to go, why or why not? So students are like, well, they could just be guessing. They could just be guessing, and they happen to get it right. So we want to know, well, what is the chance that the subject got it right just by guessing? Turns out that's 20%. There's 5 cards. Your chances of getting it right by simply guessing were just 20%. So we'll prompt the students to ask, given your knowledge of experimental design, how would you improve the experiment? So most of them will come up with the idea of replicates. They're like, well, 1 card is not clearly enough in order to show this. So now, suppose you're going to give them 25 cards to guess. So we're going to lay them 5 down, and we're going to give them a card. And now they have 5 different shapes to choose from, and you're going to give them 25 cards. How many would you expect the person to get right? Just by chance. How many, if I don't have ESP, how many do you think I would get right? So if I don't have ESP and I don't have that condition that we're trying to test for, how many would I get right? Turns out I would probably get about 5 cards right just by guessing. So we actually have a distribution, and this is where we're going to move from basic data visualization into showing how likely what we saw in our experiment is beyond that of chance. So let's continue with the cards. So what number correct would convince you that they actually had ESP? So it turns out that this is actually mini stat lesson here, that this is actually a binomial distribution because the response, our dependent variable, was yes, they got it right, or no, they were not right. So what the x-axis on the bottom represents is the number of successes, the number of cards they got right. So you'll notice that 5 is the highest peak, and 5 is what we would expect somebody to get right if they were just guessing. And and likely they could get 4 right. They could also get 6 right. But that chance dwindles. So if you were going to make a line to determine whether or not somebody really had ESP, how many cards would they get right? How many cards would you want them to get right? What about 12? Do you know the chances of a person getting 12 cards right is 0.1% if they're just guessing? So what's more likely here? That they don't have ESP and they just by chance got 12 cards right, or maybe they have ESP. So that's where we're going with our hypothesis, and that's what statistical analysis helps you decide. So typically, you'll notice that we usually make this cutoff. So let's say, for example, the probability of the subject guessing 9 or more cards right is about 4.68%. So again, the probability that somebody guesses right 9 or more cards is 4.68%. That's not a very big number. That might be a number you're comfortable with. It may not be. So this idea is we're going to say if they get 9 or more cards right, so they exhibit ESP traits. So if that's our cutoff for our experiment, 9 or more. So this is getting into a little bit of a mini stats lesson here. So typically our standard cutoff, which you've probably already been familiar with, but you may not have remembered why, is about 5%. Because 5% is our chance of saying, oops, somebody has ESP when in fact they actually didn't. Because it is possible that a person gets 9 cards right, but it's just not probable. And that's what our p-value is. Our p-value is the probability that that subject gets that observed number of cards right or more extreme, given that there's no ESP present. So in other words, the smaller the p-value, the more extreme evidence that you have against the null hypothesis. So the reason why I bring this up is every single statistical test is going to provide you with a p-value. And that p-value is going to represent that evidence that you have against your null hypothesis. So when that p-value is very, very large, you don't really have that much evidence because you're really not that far away, and it actually could happen by chance. But when you have a very small p-value, you're showing statistically that there might be a difference between the two groups. There are some debates surrounding p-value that's beyond this video that you probably are familiar with. But in general, a smaller p-value indicates that there is a difference between the two groups. There's evidence against your null hypothesis, evidence against that hypothesis of no change. So this is why we've identified all of the factors of interest and our variables that are dependent variable and our independent variables. All of those things are going to play into which statistical analysis is going to be appropriate. Either way, every statistical analysis, by and large, is going to end with a p-value. It's going to end with some number or some evidence for or against your null hypothesis. So there's actually some flowcharts in the activity handouts that will help guide your students. So if I have two groups, then I go this way, and there's a whole bunch of different flowchart options for them. If I have pre and post data, I go to one flow chart. If I have categorical, non-numeric data, I go to another flow chart. And it helps them decide what test is appropriate here, because otherwise I'm going to have to lump all of statistics into one 5-minute video. So that's not going to be possible. But what we can do is use the variables that we've already identified and the types of variables that we've identified to help us pick the proper statistical test. And there's a number of online tools that can help you do that, but they're a lot easier if you know which test to use and if your students understand how to interpret the results that they're getting. What does it mean when my p-value is very small? What does it mean when my p-value is very big? How do I interpret my results? So again, the same website, and you don't have to use this website. Again, it's free, but it offers the opportunity to analyze, for example, to compare two proportions, to compare two means, to compare two different groups. And there's more options on that website to compare more than two groups. So it helps you decide what type of analysis is appropriate for your data, and it'll allow your students to make the appropriate conclusions.
All right. Thank you so much, Jennifer Broach. I guess you'd be Jennifer 1. Because of the 3 Jennifers, I'm alphabetizing your last names. And so this concludes the recorded portion of our second day of our workshop. We have scheduled now a little bit more question and answer, and then a conclusion. Jennifer Hackney Price, has updated the website, and now there are additional tabs that have B resources as well as forensic science resources. I'm really liking the way this interaction has allowed us to give you what you need and allowed you to ask for what you needed and have us give to you. Wanted to say in the chat there, the book that Kim Kobachek was referencing is called Illustrated Guide to Home Forensic Science Experiments, all lab, no lecture. So it's there in the chat. And so let's begin with questions about this last section.
I don't have a question, but I just have a statement. Maybe this will spur some additional questions. When Jen Broach and I were talking about my experiments, my students didn't necessarily have a lot of quantitative data. They had more qualitative data. And so sitting with Jennifer and figuring out, okay, what kind of statistics do they need? What kind of statistics are they going to use? Was very helpful. and this data visualization was very helpful to them as well. So I kind of found in my experience that you didn't necessarily, or one doesn't necessarily need these really complicated statistics, which could also go to say maybe the students who are just getting their feet wet with science, with research, maybe they haven't taken statistics yet. This information that has been created within this iCURE, and then also presented by Jennifer Broach, is so helpful, hugely helpful.
Kim, I'm glad you mentioned that. And it's funny because most people and most students think it needs to be super complicated to be research.
Yes.
You know, like it must need this like really fancy. And years back, I actually brought in the lead statistician from the Mayo Clinic, and she runs all of their clinical trials. She's like the head statistician for all their clinical trials. And she mentioned, and she goes, well, I don't do anything other than a t-test and a binomial test. She goes, I don't do anything else. She goes, I spend like 1% of my time doing other things just so she can justify the need for having a PhD in statistics. You know, she's like, sometimes I just feel like I need to throw that in. So I feel a little bit better about myself, about the fact that I went to school for, 10 years after the fact that when I learned these tests. So, and we really do equate the difficulty of the stats with research, and for some reason those things tend to go together in students' minds, and to like separate that out and just make sure that because the right statistics done well is way better than some weird complicated thing that doesn't even matter and doesn't support your conclusion. So we really need to make sure that the students are aware of that aspect as well. So I'm gonna, let's see. Bill Murray's original Ghostbusters. Yeah. Yeah, Susie.
All right. We've had several people over the course of today asking about the videos. Will they be available? And so, yes, all the videos will be available separately. You can, in fact, go if you'd like to relive yesterday's workshop. It's actually all up there, all however many hours it is, on the YouTube channel, but we will be separating out the videos, and we will also be closed captioning them as well, so that those will be available in smaller bite-sized chunks. That'll probably take us a while, maybe a month or two, to get that all set up. So bear with us, but always please just email if you're like, oh, it's not there yet, what's going on? So we're here. The grant ends in August, and so this is our major deliverable. We thank you very much for coming to this, and we're hoping that after the videos get up on the YouTube in smaller chunks, that people will continue to visit them and also share them out. So we did have a couple of comments now in the chat, unless anybody had any other questions about the videos and the YouTube. All right, so the first was more of a comment about there's a major cure that has been being developed originally through the Howard Hughes Medical Institute called C-phages, and it has a discovery model, and it's found very effective for microbiology. Microbiology often starts with a discovery model, right? You know, what's on your shoe? What did your stinky brother leave on the doorknob? And so they said that found C-phages very, very effective. The students hunt for microbes and then describe them. And that is not a lot. that can be science as well, although it's a different type of science. Do you have any comments about that, Jen? No? Okay. Then there was the other one. Let's see. Should we help guide students to an experimental design that can be easily analyzed? It seems like some of the more interesting questions may need different analysis.
I'll take that. So I think honestly, the reason why most of the things are guided towards the simple analyses is because I wanted to make sure that it was accessible to all biologists. I mean, anybody teaching the cure, because I mean, as my colleagues have disclosed, I mean, their stat knowledge isn't much, I try to limit it to something that they can assist with. I also equally, I give that right back. I equally don't know anything about, know very, very little about biology. So, I mean, this is a two-way street here. So I'm not, but I try to limit it to things that the instructor could assist with. And I was just a little afraid of getting too far in the weeds beyond like a time series, something that would require a little bit deeper analyses. I don't think nothing ever forces them into something that is basic. It doesn't force them into it, but it does definitely lead them into a more basic design. The non-normal data, though, is part of the selection process, because in science, non-normal data is very, very common. And that's easily done. Actually, it's in construction in the app. It should be done in the next few months. But what to do with non-normal data is in there, like suggesting a log transform, because honestly, a log transform usually makes everything back normal anyway, or close enough. I mean, it's usually like a trial and error type thing when it comes down to there, but it is suggested in the little this basically a decision tree. Like, is it non-normal? Does the transformation work? If it does, transformation works, then repeat with the transform data, or go on to something like a Wilcoxon sign rank test, something that is for non-normal data, more ranked data. So those are options, and they actually are already embedded in the workshop, but they tend to lead to more simple designs, just so we could more broadly give this to people without assuming any statistical background. But you're always more than welcome to suggest things based upon how comfortable you are with it.
Thanks. There was another comment here, which I think is really good. A third model is a method of development or evaluation cure. For example, students compare methods of isolating bacteria. Yeah, I think that's a really good point as well. I would almost say that the mouse cure that Dr. Brownell was describing yesterday is somewhat of a cure like that, right? A development or evaluation cure. They were doing a whole series of immunology tests on wild type mice with mutations that they didn't know what the immune system was going to look like. So, that's another type of cure. I think that there's a lot of different types of cures. We had wanted to add a quantitative aspect to our cure, as well as adding an interdisciplinary aspect of our cure, because we're in a school of mathematical and natural sciences, and wanted to invite all students who wished to participate. And, we, in addition to our STEM majors, we also had other majors as well. So, I think it's been really very satisfying for me to be a part of this project. And I just wanted to thank my colleagues here on our virtual workshop who've worked extremely hard over the course of the 3-year grant and 2 no-cost extensions. One to develop the in-person workshop, and then the next one to develop the virtual workshop. And so hopefully this has been really helpful for you. We have about 20 more minutes for more conversations and more questions.
I did want to say before we break out, I wanted to say thank you to Pam Marshall for leading this project. She has been the most amazing, fearless leader. I know I speak for the rest of my colleagues that this whole project has been her brainchild from the very beginning, and she's really kept us all she keeps us all in line too, which has been amazing. And this workshop would not have been even a tenth as good as it was now. I mean, with the addition of the professional video, I mean, these were all Pam's ideas, and a herd of Jen's. Yes, and a herd of Jen's. Pam and a herd of Jen's. So I did want to say a big, huge thank you to Pam for leading this project. She's been an amazing, amazing leader. So thank you.
Gosh, thanks. I think, if we were in person, we would be hugging each other and handing each other flowers and stuff, because I know nobody who's on the video call really wants to, know that this has been a long time coming, but it's been a very long, a long process, because many of you had registered for the in-person conference last spring, and things, I mean, when March happened and we were pivoting, we were thinking, oh, well, we'll be back in person, and we'll just be back in person back in May. And then no. And then we're like, oh, let's just postpone the workshop to in-person workshop in August. And then things really started going downhill. And then it was like, oh, what do we do? So it's been a long time. All right. So we'll stop remembering that terrible past. We'll never speak of this again. Other questions about cures in general, or cures specifically? Oh, that was so nice. I'm so grateful that this was held virtually. Otherwise, I would have missed it. Yes, we are still under travel restrictions at ASU, and we still have real specific rules about how many people can meet and where they can meet. So, yeah, we're very, very grateful that you've been able to attend, and we've got lots of thanks. And so, if you want more resources, if maybe there was something that we talked about that you can't find, just send us an email. And we need to make a huge, huge shout out to the production company, ASU Productions, who was in charge of both this virtual workshop like this, as well as all of the pre-recorded videos. Without them, this would have been just us sitting around for 8 hours for 2 days on Zoom, all hating each other in life, frankly, because I've been to some of those. And so without Adam Mariner and his crew, we really, I don't think, would have been prepared to give something like this. He's just really been amazing. And so they're local here in Phoenix, a small business. And so if you need any kind of production crew, it's ASU Productions, and you can just email. Somehow we'll get a link to them on the website as well. So I don't see any more. Okay. Oh, here, can you speak to some of the outcomes you have seen from students that have taken the cures and move on to graduate school? Jen Broach.
The microbiome, not the microbiome paper but my two stat students, one of the original ones that were in the cure, did a microbiome project, and they presented it at conference, a regional conference. It was a national conference, actually, I think, wasn't it? At any rate, they both went on to graduate school. One went on in psychology and learning science. The other one went on in hearing science. I've had multiple, and then the other stat students, the one that wrote the Mind Soils paper with Dr. Ken Sweat, she ended up going on to graduate school as well. And it's funny because I think that whole project changed the course of her career pursuit, because and that one's my favorite story. I mean, there's a couple other ones in grad school, but that are in grad school, and I think they were on that path. But this particular one with the Mind Soils, it was just a class for her, and she totally changed her career. She was actually in one of the she was in the financial field, and she had this huge sacrifice where she moved away from her family. I mean, she's a single mom with kids and definitely a different situation. And she moved away from her family and did this internship because she thought this was what was going to get her. It was at Wells Fargo, and she thought this was what was going to get her this major job. And turns out she wants to do, she wanted to work with plants, and she wanted to work in science, and it's really, really, really cool. So to see how much of an impact this class has had on students. So it's really, really amazing.
And we pulled some of the data we're starting to analyze. We're trying to wait until the very end, and it looks like we have a higher graduation rate than the typical ASU student.
Yeah, by almost 9%. Yeah, the graduation rate for our CURE students is about 9% higher, which isn't typical of the population that is because we have more minorities in the class. We have more Pell Grant recipients in the class. We have more of everything that typically is an indicator of lower graduation rates. So our expected graduation rate would be a lot lower than what the standard would be. And yet our students in the CUREs are higher than the standard. So I know it's not causal. I know we're getting these students that are I can't put a causal thing. I didn't randomly assign them to taking the CURE. So I understand the ramifications of the statistics, but if you just go by what we would have expected from students that fall into these certain categories, it is significantly higher. So they are, and their responses on a graduate, we did a CURE alumni survey. The vast majority, we're talking 90% and above, say that they've used the skills later, they use them to get a job, and they use them to get either a job or into graduate school. And it was astonishing, those numbers. So we have a lot of, no obviously causal support for that, but a lot of anecdotal and convincing evidence to show that they're using this class as a platform to move on in their career and move on in graduation.
Yeah, that data just got pulled last week, and I saw it and was like, wow, we really are making a difference. Oh boy, and to teach the CURE, it's just us now, right? It was so much fun. It was so much fun, and it was so much work. But you just, when you're going through the thick of it, you just go, and then at the end of it, and then the students are there presenting their posters, and we had them do a poster and we didn't print them. They just had it on the screen and, kind of zoomed in and talked about it, and to have the students using the pronouns we and I, we thought this and we did this and we wanted to do this and this is what we saw. And every single one of those students had engaged. There was a question a while back about what to do if the students had not what to do if the students hadn't engaged and how to get them to engage. And I can't answer that question because every single one of my students was engaged. So I think we'll, wrap up here. There was something about maybe trying to get back together later on. So when we send, maybe when we send, we can send a follow up, and we can maybe put out a Google Doc and see what we can do about that. So I'll kick it on to Jennifer Hackney Price for her conclusion.
I just want to echo the sentiments of my colleagues, which is I am very thankful that you guys were all able to. It's not working.
Put the microphone closer to your mouth.
Okay, can you hear me? Awesome. So I just wanted to say thanks for attending. I think this was awesome, the interaction that we had, and I'm thrilled that you guys got something out of it, that we were able to address a lot of your questions. And again, thank you to Pam for spearheading the effort, and thank you for Adam for making this all come together in this format. So thanks to all.
All right, thanks, Jen. So we do want to remind you that we'll be sending out the surveys, so don't forget to fill out your surveys. And now we'll hear from Jen Foltz-Sweat for her conclusion.
So I'm mostly going to echo what Jennifer said, in that I'm really thankful to have worked on these cures and worked with the ASU group, because I think that it is such a valuable tool for inclusion and getting our students motivated in science. As for the workshop, again, very happy with the videos with ASU Productions. I think it was awesome, and filming the videos was a lot of fun. So definitely thank you for that, and thank you everyone for attending. Okay.
Yeah, hi. So echo what everybody else has said, but I do want to say a special thank you to Pam, because and this is why. When I came to ASU, I was coming from a city crime lab. I didn't have a lot of academic experience, and she really was my unofficial mentor. You didn't know that, did you, Pam? Surprise. So when she asked me to be a part of this grant, I was very honored. I mean, like honored, like, wow, you're asking me to do this? And so I wanted to make sure I did a good job. And I think I did. And I think we all did a great job. I learned a ton from this, not only with experimental design, but working with people, working with my students, learning along with my students. And so now that I've transitioned away from ASU back to the crime lab, I miss the teaching aspect of it. So this allowed me to continue on with that. And it was wonderful to see everybody today. And hopefully we'll still keep seeing each other. And again, thanks to Adam and ASU Productions for making everything fun. This was fun. So thanks.
Well, welcome to the end of the workshop. Of course, I'd like to echo a great thanks to my team, who made this one of the most incredible experiences. And yes, Pam's leadership was essential in making this as good as it was. And we must thank ASU Productions. But really, let's get the focus back on you all. If I may, I want to give you the most hearty encouragement possible to bring this kind of experience to all of your classes. What all of us has said is true. It's a lot of work. It's a lot of stress, because you don't know. You know, you can't tell your lab prep person what reagents you're going to need this semester. There is a lot of stress to it, but it's like a roller coaster. It's a lot of stress because it's a hell of a lot of fun. And to watch that spirit of discovery in your students, to watch their dedication is incredible. And so, honestly speaking, this is really, I suspect, what most of us got into teaching for, is watching our students having those moments of real revelation. And so I heartily encourage you with all my being, try these in your classes, whether it be a semester long or simply for one lesson. Try them because they're incredibly powerful tools of education. Thank you.
I can never follow Ken Sweat. So I think with that, we will all sign off and say thank you so much, and we will be in touch. Thank you.
Bye. Thank you.